Okay, we will go ahead and get started. Welcome, everybody. Good morning, good afternoon, good evening. Welcome to the third day of May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. We are very excited that we have this opportunity for all of us to come together and talk about important issues related to proteomics and especially computational, with especially computational focus. And we're very happy that it is possible to do this in this online format despite the circumstances. So if you are joining us for the first time, I hope that you're all healthy and safe. I hope that you can manage the time. And I also hope that these 2 weeks will be a good use of your time. So today is a very special day for us because we have two keynote presentations by leaders of proteomics, and our speakers have made very important contributions to the field. And so while most of the other sessions in the May Institute are fairly technical, where we work with specific software tools or specific types of experiments, the keynotes are really meant to highlight the applications of proteomics and the applications of computational methods and experimental methods to biological discovery and talk about the use of this technology in the broader context. So we have technically just from terms of logistics. So as you're logging in, you will see at the bottom of your screen, there are two options. One option is a chat. So chat is useful to talk to uh, presenters or to moderators about if you have a particular question, but administrative question. But if you have a scientific question related to the presentation, it is best to use the Q&A button. And so you can type your question and we will interrupt the speakers and we will voice your questions so that the questions can be answered more or less in real time. If you feel like the questions are somewhat unrelated to the presentation, maybe they will be answered in the end. So please don't hesitate to type your questions because it is also true for the speakers. It's important for them to know what is relevant for the audience. So this is the plan for today. So let me first introduce our first speaker. It is my great pleasure to introduce Professor Bernhard Kuster. Bernhard joins our program the first time this year, and we are very excited for his contribution. So Bernhard is the chair, he's a professor at the Technical University of Munich and the chair of the Department of Biosciences. So he studied chemistry at the University of Cologne, and then he had this doctorate in biochemistry at the University of Oxford. After that, he was a postdoctoral researcher in Heidelberg and also in Odense. And before joining TUM, he was vice president of CellZone. And the research that Bernhard conducts is in a very broad area of chemical and functional proteomics. And his group is particularly known for large-scale studies which characterize proteomes on the large scale. And we are very excited to hear about large-scale proteomics from Bernhard today. So I'm very much looking forward to your presentation. Thank you very much.
Yeah, thank you very much, Olga. I would have, of course, much preferred to be in Boston on site. I was actually looking forward to this, but we all know that this isn't to be. So I'm still happy, of course, to try this format. And I have to admit, I've never talked to an audience of potentially a few hundred people without seeing anyone except for myself in the camera on the top right-hand corner and Mina and Olga. But I think it's also time to say a big thank you to these two ladies for putting this together and moving it from the analog world into the internet, which achieves two things. Number one, the May Institute can happen, and number two, it reaches more people than it could otherwise do. So I'm very happy to do this, and Olga was kind enough to provide me with this title. So let's see if those expectations that have been raised with a few people already can be met. However, having said this, I'm going to also structure the presentation in a way that it's going to be quite technical at the beginning, because I do think it's important to really understand what these different technologies can and cannot measure, because that is later on at the heart of our interpretation of the experiments we do, and also the statistics or the informatics that's suitable for analyzing such data may differ depending on what we actually do. So, but before I really get going, I think it's good practice to make some disclosures. Basically, I'm just showing you that I'm a founder and a shareholder of 2 companies, and we do get some research funding from, in this case, Merck, and more research support that is more like in kind from these other companies listed down here. They didn't really have anything to do, well, some of them had something to do with the research here, but not in financial terms, purely academic. Okay, so with that, I would like to sort of pick up speed a little and essentially place TMT quantification within the broader world of quantitative mass spectrometry, because there are actually a lot of choices. And these days, maybe the most popular one are these label-free techniques, either in DIA or DDA mode, or related approaches, where a sample of interest and another sample of interest are basically in the wet lab, processed in parallel all the way down to the data analysis part only, where they're combined at this very last step. And that often times will mean that all of the technical variation that has been incurred up here is going to accumulate and be present in the data. And if there was little variation up here, then analyzing this data is going to be fun. If there was a lot of variation up there, then it's going to be less fun. Now, there's also, of course, the whole world of using standards, and they could be spiked into any sample at the workflow, either at the peptide level. For example, that would be popular for these aqua or PRM or SRM approaches. They can in principle also be spiked in at the protein level. QConCats are ways of doing this. It's less often used, but certainly possible. What is clearly the case in both of those worlds is that you have to predefine what you're going to look at. So you have to make reagents that are often very expensive to make for quantification purposes. And then there's a whole array of possible chemical labeling strategies, where we now talk about stable isotope labeling, and they all have this interesting feature that you can actually introduce labels in a whole proteome, either again on the peptide level by a chemical label, and less frequently by a chemical label on the protein. And you can, of course, also do this already at the metabolic step. The advantage of that being that you can combine your samples of interest at the earliest possible time in that workflow, and then all of the things that may go wrong down that track affect your sample and standard in the same way. So it's not going to really be a problem for the downstream data analysis. Now, important also to keep in mind is that often I get asked which of those many approaches is the best. I basically just tell people, well, depending on what you want to do, choose the one that's fit for purpose, and not necessarily the one that certain colleagues may think is the only thing to do, or that you happen to have in the laboratory. What perhaps still is true is that if you have a very long road from your sample to your data, then these metabolic labeling approaches play out their advantage. If your route from sample to mass spectrometer is short, then label-free is entirely fine. But cardinal differences in many of those techniques is whether the signal that you're going to use for quantification comes from either the MS1 level, so the precursor peptides, or the MS2, so the fragment ions, or maybe MS3, which would also be fragment ions. And there are very large differences between the two, and I'm going to cover this in a little detail. But here on that slide, I'm just giving you some idea of what colleagues have put together, which by no means is a complete list of all the chemical labeling techniques that are out there. And it's also important to note that despite the fact that some of them are quite powerful, including TMT, which I'm going to talk about mostly today, is there are a lot of little devils and a lot of little details in its chemistry, which means it's often not very clean. And so there are side reactions and what have you, and all of that is going to show in your data at some point, and whether or not it's important depends on how bad it is. And now focusing in on tandem mass specs, for those of you who may not have heard much about this, it's a chemical labeling technique, essentially that is by way of the succinimide chemistry able to derivatize the N-terminus of a peptide or the side chain of a lysine residue. And so this is called the NH2 reactive group. Then it has a little linker here, which is also referred to as a mass normalizer and a reporter group, that upon fragmentation by HCD will break this carbon-carbon bond, or by ETD will break this nitrogen-carbon bond. And it comes in a variety of flavors. So by now it comes in 6, 10, 11, or 16 plex. And what's interesting about it is you can place stable isotopes around that molecule, as depicted down here, in a way that in this case you have always 5 stable isotope centers for this TMT6 plex in each of those reagents, but just in different positions. That has the effect that the actual compound has the same mass. So we call this isobaric. However, when fragmentation happens along this bond, you can just count the number of blue stars in each of those reagents, and you will find out that there's none here, there's 1 here, there is 2 here, 3 here, 4 here, 5 here. So you can distinguish them in the tandem mass spectrum by the fact that they are all spaced by a certain amount of mass, roughly speaking, 1 Dalton. So, and that is the basis for the quantification. Okay, so and then there is higher plexing than 6-plex, as I said, 10, 11, and 16. And when you look at a prototypical spectrum of that, you will actually see that each of those channels that has a nominal mass of either 126 or 27, that many of them now come in 2 flavors. There's a split peak here in the 127 channel, 28, 29, and 30. And that split arises from the fact that you can either have a C13 compared to a C12, or an N15 to an N14, and the neutron in that chemical element is actually hasn't got the same mass, but there is about 6 millidalton mass difference. And in order to be able to detect this, you need a mass spectrometer with reasonably high resolution. About 50,000 will baseline resolve those peaks. And that is important to know, of course, when you choose the mass spectrometer for such experiments. Okay, so a typical workflow might then look like this. Important also for TMT experiments, you have to pretty much define a fixed number of experiments you want to compare later on. That's not entirely true, but most of the time people do this, and you could have, say, 6 or 10 or 16 dishes of cell culture, and then you basically want to treat it in 16 different ways. You do that, you harvest your cells, you digest the proteins into peptides, and at the peptide level, you introduce your different TMT labels, then combine all of the samples and do a combined LC-MS-MS analysis, where your MS1 spectrum will have a lot of precursor ions, and then you select out, or the mass spectrometer selects out 1 for MS-MS, and you get your fragment ions. And importantly, now at the low mass end here, you will find your TMT peaks, and they will split up according to the plexing level that you've employed. And then importantly, the quantification of these 6 or 10 or 16 samples is going to happen relative to each other, and not relative to some spike standard or some such. So it's always a relative quantification. So another cardinal difference between a TMT-based experiment and, for example, using XIC, so extracted ion chromatograms from label-free or DIA or even PRM, is that XICs integrate either the MS1 or the MS2 signal across the chromatographic elution time of a peptide. TMT does not do that. TMT captures the MS2 signal intensity at a certain point in time. And that is largely arbitrary. So we don't know if we're going to, in TMT world, pick the chromatographic elution time at the apex, or early on, or late, or somewhere in the middle. So that's very often not really within our control. And that's an important difference, because we use, in this case, the integrated intensity across the LC peak, and in TMT, we just take what we get at that moment in time. And that has some consequences. Okay, also a word about mass spectrometers, because they are still sort of ordinary detectors, quite powerful, still ordinary detectors. So many things apply that apply to any other detector. First of all, one has to realize that any quantitative analysis tends to need better data than qualitative analysis. But more importantly, detectors all have a certain range within which they can measure properly. If your signal is great, like you have an isotope cluster here of a particular peptide of something like 1817 70 Daltons. You see all the isotopes, they are nicely resolved and they follow exactly the isotopic pattern that one would expect. However, if you had very, very high signal, you may actually saturate your detector and then your signal might be squared off. And you can imagine that this would be detrimental for the quantification. Likewise, at the other end of the intensity scale, you might be losing sort of your peak in some, not necessarily only noise, it could just be noise. So when the signal is very low, or there could be other interfering peaks here, which makes picking the right isotope cluster in here not can make it difficult. So we basically need to keep in mind that if you have saturation of your detector, then the quantification is going to be wrong. If you have a too low signal, that may prevent detection, you're going to create missing values. Or if you have a low signal to noise or interfering signals, you get false measurements or false quantification. And what we call the dynamic range really is that difference between how little can I detect in the presence of how much of something else, and that will also have an impact on the quality of your quantification. So that's
there's
true.
A question.
Yeah.
Can TMT label be done at the protein level as opposed to peptide level?
Yes, it's possible, but it's very rarely done because when you want to label native protein, you have to do this sort of in an aqueous buffer system. A lot of the residues you may want to label might not be accessible. So you get a lot of variable labeling on different sites. You may actually want to do this because it may tell you something about whether those places on the protein are surface exposed or not. But it's quite rarely done, but it's possible. Okay, so more words about MS1 and MS2 signals, just to keep in mind. Usually MS1 signal intensity, so the precursor, peptide precursor, is usually much higher than the signal in MS2. However, and that you can see from this ion chromatogram up here, these are all the precursors that hit the detector at any one given time in this chromatogram. And if you strain your eyes, you can see that this number is higher than that number. The bottom one is, in this case, a MS2 trace for the ions in all the tandem mass spectra over time. And you can see it's much more spiky. And it kind of is cleaner. It's often considered cleaner. It has lower noise or fewer interfering signals. So, but these are important differences that are going to have consequences a little later. So now going into the trenches of TMT, you may or may not know that by any stable isotope label are rarely or never 100% pure. So there are extra peaks in the spectra, both in MS1 and MS2. So if we look at our precursor ion here again, it has that nice cluster of isotopes, but there's also one signal at -1 Dalton. And what that is simply that one of those carbon-13 or nitrogen-15 atoms isn't there. So the purity of the isotope label isn't 100%. And when you buy TMT reagents, the vendor gives you a wash list, how we call it, of how much signal or how little signal you should expect at the -1 position. And then, of course, this is not only true in your MS1 signal, but if you fragment all of this, then this isotope impurity will also show in the MS2 spectrum. And of course, also your TMT reporter ions will also generate a C13 satellite ion, and that has to be corrected for. Now, there is uncertainty in the isotope impurity correction in that list. So that's usually what people don't necessarily think about, that it is actually there. And the accuracy of determination of this isotope purity is actually not much better than 10 or 20%. So when you now take any TMT signal that is uncorrected, here is a channel 126, 127, 28, 29, 30, 31, you can actually see that there is at the -1 position, there is some signal. It's not a lot, it's just 0.2%. But when you then correct it, it should be gone completely, but that doesn't sometimes happen. So sometimes there's residual signal even after the correction. And if you look at the green channel here, that's the uncorrected data, and here's the corrected data. You can see that some of those numbers are now closer to 0, but there are some that are still not 0. And it could be different for each channel. And that has, again, consequences we're going to see in a moment. Yet another thing that most people are probably not aware of, and often it's taken care of by software these days, but also an ion trap detector can be saturated. And people around Markus Banschew have noticed a while ago, or it's already a few years ago, that if you put too many ions into a C-trap or an Orbitrap instrument, then you get space charging, and that can lead to what is called ion coalescence. So here we're looking at the heavy and the light channel of the 130 TMT reporter ion, but if there's too many ions there, they can coalesce into a single peak, which means this quantification of this peak isn't somehow representation of those two, but there's no way of knowing whether this is a 1-to-1 mix or whether it was a 9-to-10 mix or a 1-to-10 mix. We don't know. And that could happen to various degrees in the different channels. And one tries to overcome this or prevent this by making sure that one never allows too many ions into the system. Okay, so let's say we have all of this under control. You can get some idea of what the dynamic range of your quantification may be. And here we did a couple of experiments a while ago where we just took HeLa digests and diluted them separately and then started to mix them together in various ratios. Because we always use the same digest and we dilute them, we kind of wouldn't expect any sort of what is called ratio compression. I'll come to that in a moment, because every peptide is diluted by the same factor. Now, but if we do this, and we now look at this plot down here, what you can see in this plot is that when you mix TMT channels together that have different quantities of digest in them, you can calculate, of course, what you would expect in terms of ratio, and this is in a log 10 scale, and then you can measure what you actually get in the experiment. And in this case, when we just look at the 200 most abundant peptides, you can see that while we're diluting the sample, the signal goes down, and your expectation, the expect ratio of the 1-to-1 versus 1-to in 1,000 mixture is actually quite good. So goes linearly down, but then when you go to about 1 in 1,000, you hit the bottom. So which means when you dilute the sample further, you don't further decrease your signal. Okay, so clearly we've reached the end of the dynamic range here. Now, you can do a second experiment where we now keep every other TMT channel empty, and again, analyze the 200 most abundant peptides. And then you see something perhaps surprising, that while the normal dilution channels that actually have digest follow exactly your expectation of going down and down and down, with the expected and the observed ratios being the same. However, in the empty channels where we didn't even put any peptides in, we begin to see signal. And this could be as much as 1 in 100 or down to 1 in 1,000. So our effective dynamic range that we can actually measure is decreased to 1 in 100 or to 1 in 1,000. Now, and if we now take all TMT channels and occupy them with sample and dilute, and now we can look at, let's say, the top 10 most abundant peptides in that sample. They again are nicely behaved along this axis, but you'll quickly see that even when you look at the top 100 peptides or the top 1,000 or all of them, you can see that we are populating here signal in dilutions that we really shouldn't have any more signal. And it could be not as bad for the intense peaks. It's still 1 in 1,000 dynamic range, perhaps. That's the green dots. But it could be as bad as 1 in 20 for the less abundant peaks, like on this blue thing here. And if you look at the whisker up here, this is not much more than 20-fold, even though you would expect that to be a 1,000-fold. So, and then similar argument applies to that if you actually hit 0 and have missing values in your TMT experiment simply because there wasn't enough signal, then you can, of course, impute values. But if you do that, you have to be aware of the fact that if you impute values for peaks that are usually very intense, again, the top 200 peptides in these dilution experiments, this is fine. You can impute, but then you still hit a bottom at some point, and that bottom that you hit is actually quite close to the 1 in 1,000 we had over here. But if you look at the least abundant peptides, then you're hitting that already at a factor 1 to 10. So all of that means is your quantification results are also going to be influenced by what your detection system can actually measure. So be mindful of that fact when you look at TMT data. Sometimes it's not a problem. Sometimes it's a terrible mistake.
Bernhard, could we take a few questions? There are quite a few.
I imagine, yeah.
I think we will not be able to answer all of them, but let's just do a few. So can you comment on label-free versus TMT in terms of dynamic range?
Yeah, that'll come in a few slides if I can hold that off for a moment.
Sure thing. And so then how can we reduce or avoid reporter ion interference?
Yep, that's also it's a very good question. And one easy way of doing this is by adding another step of MS, which is called MS3. So you're selecting your fragment ions again in your trap, and then you're fragmenting those fragment ions to create reporter ions that you then measure sort of in an MS3 level experiment, and you can reduce much of that interference.
Another question, what is the usual amount of ions safe to use to avoid space charging in orbit traps?
Yeah, so
in orbit trap size is determining the amount as well.
Yeah, well, the capacity of the trap is determined by many things, but if you want to play it safe, 1E5 ions is what you can put in usually.
Another question is, if you have an empty channel and if they're not always to be zero, how to characterize the labeling efficiency, and what kind of percentage or intensity is tolerable?
Well, the labeling efficiency is actually usually best evaluated either at the MS1 level or actually at the level of a search engine. When you go searching with TMT labels either as a variable modification, and then you can just count the number of peptides that only have 1 or 0 labels, and you can look up their intensities and build essentially the ratio of the intensities with a label and without a label, and that gives you a broad sense of the labeling efficiency.
And maybe the last for now, what software do you use for MS3 level analysis?
Most of the time we use MaxQuant.
Okay, thank you.
Okay, so after all this, we're going to look at one other thing, that a term that many of you will have heard about, which is called ratio compression. The examples I showed you previously ideally wouldn't have, as I said, any ratio compression because it's all the same peptides, just diluted at the same relative ratio. So we shouldn't see any distortion in those ratios. However, in practical terms, there is this phenomenon called ratio compression or ratio distortion, and this really arises from the presence of other peptides at the moment the mass spectrometer decides to pick one precursor for fragmentation. So we can take a look, for example, here you have this black isotope cluster here for one particular peptide, and there's some red signal here, but it's at a very low level. And if you then do MS, your reporter ions are going to hopefully mainly represent the black ions here, and the red ions might be representing a smaller amount of the signal. So the contribution of the red ions to the black signal isn't going to be very high. So therefore we can expect that the ratios that we're going to compute from one TMT channel to the other are going to be relatively close to what the actual ratios are. However, just a word of caution, we of course have absolutely no way of knowing if these interfering peaks are going to produce, like I depicted here, a 1-to-1-to-1-to-1 ratio, or if they themselves have ratios in that experiment. But as a simple sort of matter of principle, if you have low interfering signals, you usually get little ratio compression because the contribution of that interfering ions here to the total TMT signal here isn't very high. However, if you have more interference, and it happens more often than you think, then you could actually have a much bigger contribution. And again, if we assume that the red ions here are all 1-to-1-to-1, you can actually see what you measure is the total signal, but your actual ratios of your black things would have been much bigger than what you can actually measure. And this is why it's called compression, because the true ratios are always appear much smaller than they truly are. And this would lead to actually quite a bad accuracy. And accuracy being defined how close is my measurement to the actual true value, whereas precision just refers to how reproducible my measurement is, no matter how correct it is. So, and on the right-hand side, and where this really shows is when you start mixing different things together. So in this experiment, for example, we took, I think, 10 or 11 cell lines, digested them, labeled them with, where's my mouse? My mouse is gone. Labeled them with TMT, and then ran them down different types of LC-MS. And we then asked the question in this plot is how many of those peptides show kind of what range of TMT quantification, and that's a log2 scale here. And you can see that no matter what we do, the median or the sort of the majority of all those TMT reporter ions have ratios that are not much bigger than 3 in log2 world. So that's a factor 8 or maybe 4. That would be a factor 16. Some of them extend all the way up to a log2 of 10, which is roughly 1,000-fold, but only very, very few. And to the question that just got asked, so what can we do? So if you look at the 4 lines, the first one being a nano LC and MS2 readout with a short gradient, then our range of dynamic range in which 95% of all data is just 1 in 50. We can do a better chromatography, then it may just increase to 1 in 75 or so, simply because not as many peaks are eluting at the same time. But we can then also do the MS3 experiment that I alluded to, and then it goes up to 1 in 100, and we can then extend again the gradient for the LC to simplify the mixture, and then we can go up to 150 in this range. And here, mind you, this is all exactly the same sample. Okay, but your quantitative dynamic range is quite different depending on how you analyze it on the mass spectrometer. So that's important to keep in mind. Okay, so for some reason I cannot get my mouse back. Can I have a quick?
It is back.
You can see it. Okay, yeah. Very good. Okay,
and
so
maybe can we do a couple more questions as well? So yeah. Ratio compression may be high for one peptide, but low for another peptide. Is there a method to correct for quote unquote noisy peptide?
Yeah, so there are approaches to do that. I mean, there are some software tools that can actually look at that window that got isolated and then ask how much of the total signal in that window can we attribute to the black or the red signal. And then this is applied to correct this. However, it works under the assumption that most peptides are not changing in an experiment, so that they would produce 1-to-1 to 1-to-1 ratios. And then this is a way of how one can clean this up. But as I said, it is an assumption that may or may not be always justified. But generally, this works, and it improves matters. And mind you, in the plot on the right-hand side, we've already done that.
And another question is, what is a good cutoff for interference?
I don't think there is really such a thing as a good cutoff, because it is a continuous scale. It's hard to know. What we generally observe is the more peptides for a protein we have, the better the quantification becomes, because you can either exclude the weird ratios completely, or if they have, let's say, not much intensity, then they also don't contribute so much to the total signal. But there's really, it's hard to deal with this post data acquisition. A very new way of dealing with this has been published also by Steve Giggy's lab, where you're on the fly trying to identify a peptide from MS2, and then you only do MS3 on those peptides that you've already identified, and then that actually produces very good results in terms of removing ratio compression issues.
So a couple of technology questions. Why high flow rate is giving better dynamic range than nano?
Yeah, so if you go to higher flow rate, we just do better chromatography. For those of you who look at your nano LCMS chromatograms, sometimes you're shocked and sometimes you're in awe. Often, essentially, these very narrow columns, 50 or 75 micron, they don't have a lot of capacity. So even the slightest overloading of the column will produce broader peaks. And broader peaks in chromatography means more precursors present at any one time. And if you go to higher flow rates and bigger column diameters, simply your chromatographic peak width gets smaller and smaller, so you're reducing the complexity of precursor ions that fly into the mass spectrometer at any one time.
And just two more technology questions. With low sample volumes, is high flow LC possible?
With low sample volumes or quantity, you mean? Maybe quantity is what is meant.
Probably.
Yeah, so higher flow rates and bigger columns usually eat some signal. At the same time, the chromatographic peaks tend to get much sharper, so the concentration goes up. And because electrospray is a concentration-dependent process, some of it can be compensated, but only within limits. So the highest sensitivity will be achieved in nano LC, and when you go to higher flows, you will compromise some sensitivity, But what people are observing when they go from 300 nanoliters to 3 microliters a minute, they don't really lose a factor 10, which you should in theory lose, but they only use maybe a factor 1.5 to 2. And when you go to much higher flow rates, even like 100 times higher than the typical nano, what we observe in our lab is that our sensitivity only goes down by about a factor 5 to 10. And for many proteomic applications, this is not a problem. For some proteomic applications, it's a complete killer and you can't do it.
And the last question for now. So can you comment on HPLC versus ion mobility fractionation?
Yeah, well, that's an early, let's say an early field. Of course, I would never compromise LC quality for anything if possible, but if you have to, then ion mobility can compensate to some degree. What we find is that if you call it if one would want to call it the peak capacity of ion mobility is smaller than that of chromatography. So therefore usually gain actually faster if you do better chromatography, but that may depend also whether you are dealing with a Bruker times type of ion mobility or Waters type of ion mobility or Thermo type of ion mobility.
Okay, thank you. There's a lot more questions, but
Yeah,
let's move on and maybe we can answer towards the end.
Yeah, so there's a reason why I'm dwelling so much on the techie things, because TMT, we like TMT a lot and other people like TMT a lot, but it has some sort of nasty side effects. And one is shown here. When we look at tandem mass spec use and reproducibility, what this ratio compression issue can do and will do is it leads to a great apparent reproducibility between your experiments. So let's say you have done a TMT 10-plex experiment and you have triplicates of 3 experiments in here to occupy 9 of your channels. You can, of course, ask the question, how do my TMT channel intensities correlate with each other? And if you have replicates, you can ask the question, how do the replicates correlate to each other? And then if you get a very high number, you may be happy because you say, I'm very reproducible at what I'm doing. And this is probably interesting for the stats people. So if we look at this particular example from a TMT 10-plex experiment where we only used 9 channels, but here's 3 replicates that you can correlate to each other. Each replicate also had 2 different conditions. That doesn't matter for now, but look at these Pearson correlations between those experiments. They are near perfect. And if you use coefficients of variation for this and plot them out, then it turns out that in this data set, on the level of phosphorylation sites, we get 12% CV on 10 or 20,000 phosphorylation sites. That's pretty good going. That's better than any PRM I know. Or if you roll up the TMT intensities of peptides into proteins, the median CVs are down to 2%. So much of that is driven by ratio compression, because if your signal is compressed to 1 to 1 to 1 most of the time, then obviously you're going to get a very great apparent reproducibility, but please don't interpret that such that TMT has great quantitative precision. And that is what I see a lot in the field, that TMT is sort of equals great precision, and to some degree that is true, but a lot of that precision is only apparently so because of ratio compression. Okay, so the question was asked, so how do the 2 levels, MS2 and MS1 or MS3 and TMT and label-free compare? This is what is shown in these plots. And one thing, again, to keep in mind is that MS1 peptide detection is actually most of the time limited by signal to noise. There's usually a lot of signal, but if your noise level is high, your signal to noise is low, and then you may not be able to quantify well. MS2 or MS3 peptide detection or fragment ion detection is usually limited by the actual signal. Do you have enough signal? Then you get nice measurements. Don't you have enough signal? You get poor measurements. It's important to remember that both of them have a finite dynamic range. And this is what is shown here on this plot. Here, this distribution is TMT reporter ion intensity on this axis. And you can see it spans about 4 orders of magnitude if we look at across all LCMS runs, I should say. And here the equivalent actually label-free, it's on the very same data. You can see it has this distribution, and these 2 distributions are quite strong, far apart from each other. roughly by a factor of 1,000. And that reflects the amount of signal that is in your detector. And you can see another thing is that here on the label-free MS1 dynamic range, you can see that this side sort of looks like a nice right side of a normal distribution, and then it sort of crashes down on this level. This is where the background signal kick in, not necessarily noise in terms of electronic noise, because they have very little of it, but other stuff that is eating the dynamic range of your detector. In MS2 or MS3, you have a lot less signal in total, so you have a lot less problems with signal to noise issues, and there the distribution is actually a bit smoother on both sides, or more symmetrical. So what that means is some of your quantification results in MS1, if they are down here, could actually be distorted, whereas that shouldn't happen so much at the level of TMT in MS2 or MS3. Now, if we roll that from the MS intensity to the peptide dynamic range, we can actually see that for TMT and label-free, it's actually not so different. And the median is actually at about a factor 32 for label-free or 37 for TMT, so not really much difference. But if we now roll up the peptide intensity information into a protein quantification, then we see that the label-free has a bit of a larger dynamic range than the TMT, and that is simply because we can add up the ion intensities of MS1 signal in a way more efficiently than we can do that with TMTs. So peptide level quantification is just as TMT, just as good as MS1. Protein level has a bigger dynamic range in MS1. So another point that is very often discussed, and I therefore want to cover this as well, is the missing value issue. One of my favorite topics. When we do single TMT experiments, we observe that we have typically very few missing values. But I would argue, or I would like you to think about whether they are not missing for the right or the wrong reasons. And what you see in this plot down here is actually 55 lung cancer cell lines that we've analyzed, either by label-free quantification, that's the left sort of block, or by TMT. And we had to use some 14 or so TMT experiments in order to cover all 55 cell lines. And then for label-free, of course, we could analyze them each separately. And when we look at the number of peptides or proteins we observe in the label-free, we get in total about 9,000. In the TMT, we get a total of about 9,000. But if we ask the question, how many of those proteins are actually detected in each and every single cell line, then the label-free comes back with about 3,500, and the TMT comes back with about 6,000. And the green bars are just those proteins that are detected in 9 to 54 cell lines, and the blue that you can probably barely see is that these are proteins that are very rarely detected. Now, from this one might conclude that TMT doesn't get in total more protein in a single experiment, or even when we put all experiments together, but it seems there seem to be much less missing values in the data. But if you now think back to what I told you about ratio compression, this may have a different interpretation. So at some point, the ratio compression that we have in the data may falsely suggest that we have a protein identified, whereas it is actually not present. So how can we get an idea about whether this is true or not? Here's two plots that are essentially showing the same data. On the left-hand side, essentially on the x-axis, we plot the difference or the pairwise comparison of LFQ intensities of a protein in any two of those 55 cell lines. And on the y-axis, we do the same thing for the TMT channels. And there's already two things become apparent. If we look at the x-axis, the dynamic range of the LFQ is bigger. It goes from -6 to +6 in log10 world, and the axis on the y-axis go from -4 to +4, still a very wide dynamic range. But we can also see that the correlation is not following a slope of 1. So something is different, and it's maybe easier to see on the right-hand side of the plot when we bin now those pairwise comparison differences in the LFQ world on the x-axis and plot them against the actual fold difference. And then we can see that the gray boxes are all those proteins that are within a certain bin. And you can see they follow this linear trend. And in blue are the same proteins, but in the TMT world. And you can see that they have a very different slope. And they also have many, many more sort of much more spread around the medians. And this is purely a result of ratio compression. And therefore, the reduced number of missing values in TMT experiment actually primarily are a result of ratio compression and not of actual measurement. That's important to understand, and it can be getting very bad. I unfortunately couldn't quickly find the example that I wanted to show, but imagine you have an experiment where you have a protein in one sample and you have a knockout in the other cell line. The ratio should be infinite, and we've done such experiments where we compared knockout cell lines versus wild type, and in the most extreme case that we encountered, the knockout cell line only showed a reduction of protein intensity for the knockout gene by 50%. And the other 50% of the signal had nothing to do with this peptide or protein. It had all to do with ratio compression. So it can get very bad.
Bernhard, could you take a few questions? There is a lot of questions already.
So
one question is it recommended to compare TMT and LFQ as a quality control check on a regular basis?
Yeah, that really depends on what you compare it. If you compare it really on very complex samples, then the comparison always says the correlation is there, but it's not particularly good. Again, that may not mean much for your interpretation of your results. It may just mean whether you're looking at accuracy or precision or what is more accurate than the other. If you, however, use very simple mixtures to ask that question, it should be fine.
So a clarification question, the data you were showing, are these MS3?
The TMT data that we are showing here is actually MS3, yes.
Okay. Another question, how does TMT perform on highly complex samples with large dynamic range, for example, plasma? Is it a large improvement compared to LFQ?
No, it's actually a deprovement, if you like, because the ratio compression problem is going to kick in big time. So I'm going to come to that. I would not really recommend using the TMT for body fluids.
Interesting. Another question, when you have multiple TMT sets for different cell lines, do you search the data all together in MaxQuant? And how do you correct using the pooled sample?
Yeah, so I would recommend searching them together, because only then can MaxQuant make sure that the protein grouping works. So if you search them separately, that is one way of getting more proteins, but that may just falsely happen because you want to make sure that all your peptide grouping, protein grouping procedures are not impacted.
Another question, can the reduced missing values in TMT be the result of using a reference standard sample included in each TMT plex?
That's one of my favorites. I didn't mention it yet, but all of what I said, of course, is highly important for the single cell guys and girls. Because if you're using a reference spike in, if you will, or channel, then depending on how much of that you spike, this is what you're going to measure. And you're going to measure all the other channels relative to that. And if that one channel that you use as a reference has 2 times, 4 times, 10 times, 100 times more sample, then most of what you're going to measure in the other channels, or more and more of what you're measuring in the other channels, is going to just be spillover from the reference channel. So that is a difficulty. But yes, when you have to combine many TMT experiments into a larger analysis, then you should actually use one, better 2 reference channels.
And one more, in TMT experiments, quantifying more PSM per peptide and protein, does it lead to better quantification at the protein level?
It does, but not quite as strongly as for label-free. So it's actually interesting how stable TMT peptide level quantification can be, and that depends a bit on, and really that depends on how much sort of ratio compression you have and how much signal you have. But it's actually interesting to note that the improvement with TMT, or it saturates rather quickly, or it stabilizes, I should say, it stabilizes rather quickly. If you have 3, 4, 5 peptides in a TMT, your quantification usually stabilizes very quickly.
And maybe the last question for now, do you think a narrow isolation window, such as 0.5 m/z, would help to minimize ratio compression?
It certainly helps. People have shown that. It minimizes, or it makes the problem smaller, but it doesn't remove it completely. Also, when you do MS3, people have experienced that it makes a difference whether you pick 10 of your MS2 fragments for further fragmentation by MS3 or 1. So the least ratio compression you get is actually when you just pick a single fragment ion from your MS2 and do MS3 on it. But of course, the cost of that is very apparent. It is going to cost sensitivity. So in the end, you're going to want to operate a mix of good LC, narrow isolation, and if you can, the computational sort of cleanup of the data.
And maybe the very last one in this batch, do you need any change for a 16-plex TMT, change in the protocols?
In the labeling protocol, not, but what you sort of have to keep in mind is that the capacity of your LC-MS system hasn't become bigger, but you're now pooling 16 samples together instead of, let's say, 6. So all of the available measurement capacity is going to be taken up by all of those ions. So you might actually expect that the minor components in your mixture may be harder to detect, simply because the major components are eating up all of the available space.
Okay, thanks a lot.
Okay, so this is sort of the end of my technical part, and I hope we've been already using a lot of time on questions, because otherwise I'm doing very badly on the large scale stuff. But at least some of the data that I showed you is actually based on pretty large data sets that allowed us to sort of have a statistically sound evaluation of such data. But if you want to at least take all of that with sort of the disclaimer that these are the things that I think and not necessarily others. But as I said, we actually quite like TMT labeling, and there's a couple of clear advantages. And one is, I haven't talked about this, but you can apply it to any and all biological samples you can think of, at least in principle. And you can multiplex. So measuring 16 things in one go clearly has a lot going for it. And interestingly, and pragmatically, the quantification results are rather less dependent on your LC performance than label-free experiments. So if you're into PRMs, for example, you really want to make sure that your LC performs the same day in, day out. If you do TMT, that doesn't matter so much. This is, by the way, one of the reasons why some PIs prefer using TMT labels even to measure patient cohorts, for example, because they don't have to worry so much about the performance of the LC. Now, we talked about no missing values in inverted commas. So TMT does have, in fact, reasonably good precision. And well, the tag also to some degree makes the peptides fly a little better. The downsides of the technology we've also looked at is co-fragmentation can compress ratios, leading to underestimation of the true values. So that means poorer accuracy. And the limited signal in MS2 may compromise sensitivity. And we usually in TMT, because of multiplexing, see a little less IDs than in label-free, but that is not really a big problem. And the last point I alluded to is TMT labeling happens relatively late in a workflow, or oftentimes, or not always, but sometimes. So all of the technical variation that is accumulated up to that point will be in the data and hard to remove. So when should we use what? Now this really becomes entirely subjective. This is why my first bullet is really don't be too dogmatic about it. Think about what is important for the experiment that you do, because sometimes quantitative accuracy is more important than precision, or the other way around. A lot of times you can completely get away with good precision, not worrying too much about whether your quantitative results are actually true. We think it's a great tool for multiplexing experiments that happen within the same biological system, where often, not always, where often precision suffices. So if you run time courses or differential treatment on the same cells, or biological systems, or protein turnover. I may get a chance to talk a little about this, or this thermal proteome profiling that many labs are fond of doing these days. Where I think it is more challenging, but by no means can be done, but it's just more challenging, is when you're multiplexing samples that come from very different biological backgrounds. Because I would argue you often need the accuracy to really reflect in your quantitative data those differences in biological systems. So panels of cell lines and tissues are a bit of a tricky business, or cohorts of patients, and that question got asked, or anything really where you have to expect very large changes, because very large changes are often harder to analyze by TMT. And what do I mean by large changes? Anything bigger than a factor 20 or 50 or even 100. Okay, so that was the techie part of it. And now I was coming to a few examples of where we're using it. And Olga, you need to sort of tell me how much or how little time I have. I think 50 minutes of our 90-minute slots are up, correct?
So I think you can have 35 minutes more.
All right. Anyhow, so you basically stop me. So the first example where we used TMT a lot is thermal stability of protein profiling. And one question as a dad of 3 kids has always sort of kept me wondering when there was another cold that came along with fever, is why do humans actually die if our core body temperature rises above 42 degrees? And some time ago, a postdoc in my lab did a brave Bradford assay experiment, basically taking some lysate of cells and just heat them to temperatures between 30 degrees and 100 degrees, and each data point here is one of those experiments. And you can see that when we look at the y-axis is the amount of soluble protein after heating, is that it follows this sigmoidal behavior where starting at about 42 degrees, we are seeing a rapid loss of protein due to precipitation. And this is when essentially, if that happens inside your cells and proteins begin to precipitate at a massive scale, then those cells are certainly going to not be happy. And actually, hyperthermia is characterized by multi-organ failure because the proteins inside the cells are actually crashing. But it's also interesting to see at the other end of the distribution, at the high temperature range, there's about 20% or 15% of the proteins that at least remain soluble. They may be completely denatured, but they don't crash out of solutions. Okay, so this seems as a characteristic of proteins, that they can aggregate or co-aggregate with other proteins when heated. And we set out on an experiment where we asked the question, what actually determines this thermal stability of proteins? And in order to do this, we did a reasonably large experiment, essentially where we looked at 13 different species and heated them up to different temperatures, and basically leading to protein precipitation to various degrees. And we encoded each of the temperatures with a TMT label. And then using the TMT signal, we would basically build melting curves for all of those proteins, for all of those species. And in total, we did about 77 of these experiments, and covering 140,000 such curves, and representing about 48,000 different proteins. And we hoped with that data set, we might actually learn something about that question, what determines protein stability. And here are just examples for these melting curves. And here TMT really was crucial for us, because we encoded every temperature with one TMT label to be able to create these curves with good quality. And you can see here that this protein IK red shows a melting characteristics where half of it has precipitated at around 46 degrees already, whereas this other protein printed in black called MAPT or tau, it doesn't really melt at all. It may well denature, but it doesn't crash out of solution. And so one thing that we clearly learned very quickly is that the melting characteristics of these proteins can be very different from one to the other. And we just, for practical reasons, classified those proteins into what we call low melting points. So where the area under the curve of these melting plots that we have at the bottom here, if the area under that curve was less than 35%, we call it a low TM, so an early melter, if you like, or if it was between 0.35 and 0.65, it was a medium melter. High melters would be characterized by an area under the curve bigger than 0.65, and there would be no melting, non-melting proteins that actually never reach 50% reduction, like here in this case, this tau protein. So that's just a pragmatic way of categorizing the proteins. It's rather arbitrary, but that's what we did to try and ask a couple of questions, and that's going to be coming back to this in a few moments. But one of the things we saw is we actually summarized in this plot when we look at all these different organisms, and they are now organized by the actual optimal growth temperature of them. So at the top is this bacterium that loves living at about 70 to 80 degrees near thermal vents in the ocean, and all the way down to this bacterium, which is called something Antarctica, which is found in the waters of Antarctica actually, and is happy down to 4 degrees, but optimally grows at something like 15 degrees. And you can see that all those organisms show some distribution of the melting points of their proteins and commensurate with the temperatures at which they live. Obviously, the Thermophilus has to find a way to make functional proteins at 70 degrees, otherwise it couldn't live there. Likewise, at the other end of the spectrum, that other bacterium that likes cold water apparently doesn't have to make particularly cold adapted proteins because when you look at the distribution, it's not really much different to C. elegans that lives at 20 degrees, or even Arabidopsis thaliana that lives at about 25, or the zebrafish that lives at about 30 degrees. So one thing that is noteworthy in this data is the open circles. The open circles denote the optimum growth temperature. And what you may be able to see is that at least all of the higher organisms, the Arabidopsis, also the C. elegans, the Arabidopsis, the zebrafish, the fly, humans, mouse, they all have a gap between the optimal growth temperature and the left side, so that's the cold side of the melting point distribution. And we think this is, whereas the bacteria don't have that. The optimal growth temperature is very close to the beginning of where proteins actually start melting. And we think that has something to say about the biology. So these higher organisms have to operate some safety margin because they live in environments that have changing temperatures. But interestingly also that zebrafish, it lives in warm waters, and it's sort of a endless water bath and constant temperature. So it has a reasonably narrow range of melting points because it doesn't really have to optimize this. Whereas the bacteria seem to be all sitting in a niche where they've adapted to these temperatures, but then the moment you take them out of their temperature, particularly on the higher temperature side, they don't like that at all and actually die. So we thought that was an interesting observation in the data. And then we did a series, what I still think is reasonably elegant experiments actually, and asked whether the thermal stability of proteins is an intrinsic property of a protein. So what you see here is a series of 3 experiments where we compared the melting behavior of proteins in this thermophile, Thermophilus versus E. coli. And on the left-hand side, you can see that if you prepare lysates, or if you heat up cells, these are the scatter plots that we plot, you can see that the E. coli proteins on average have melting points of some 50 degrees in lysates and 52 degrees in cells. And the Thermophilus has the equivalent of about 85 degrees. So far, so good. And then we asked, okay, what if we mix those lysates together? So we prepared separate lysates of these bacteria, then mixed them together, and then heated the proteins as a mix. And when we then look at the correlation plots of whether those proteins, of what the melting points are, if either in the individual lysates or in the mixed lysates, we actually see that these proteins from E. coli don't really care much about the presence of the proteins from Thermophilus or vice versa, indicating that just the mere presence of other proteins doesn't really play a role. And then we took about 70 genes from Thermophilus and expressed them in E. coli, so that the host organism now was in both cases E. coli. And then we compared again the melting temperatures in cells And then we found out that no matter what we did, the Thermophilus proteins always had high melting points and the E. coli proteins always had low melting points. So from that, we concluded that the thermal stability of these proteins is primarily a protein intrinsic property. Well, one would have maybe guessed this from the start. But then when we looked at this a little more closely, we also found that the environment in which these proteins are also does play a role. So here what you see is maybe 24 cellular proteins that we also happen to find in a couple of human body fluids. And the open circles are the melting temperatures or the AUC, area under the curve values in the cell lines in either HEC293s or in hepatocytes. And you can see that in the colored dots, the AUC values were always much higher in those body fluids. And the interpretation of that is twofold. Number one is you're actually diluting, compared to a cell line, you're diluting out protein, so it can't really find other proteins to precipitate. But it also says that the protein concentration in the cell, in this body fluids are also generally lower than that in the cells, so that it is harder for those proteins to find others to co-precipitate. So that's, I think, an important learning that comes out of this, because it, sorry, I have to stop my timer here. Because it says no protein sort of aggregates and crashes out of solution alone. It needs other molecules around it that have a similar behavior. And where this is very clearly the case is when you look at the composition of protein complexes. And on the right-hand side, there is a protein complex from a zebrafish, and each of those yellow lines is a subunit of that complex, and you can see that they all co-aggregate and then crash out together. And then the equivalent complex on the human has a few more subunits, but it essentially does the same. So it appears that protein complexes, this is also what other people have seen, of course, before, but it seems that this is also kind of conserved even from one species to the other. Again, noting the important biological importance of protein-protein interactions and regulating cellular activities. All right, so then on our sort of voyage of trying to understand what the thermal stability of proteins may be driven by, we looked at a lot of things, and it became rather frustrating because even though we found a lot of associations, a lot of them were really, despite being statistically significant, not particularly telling. So on the left-hand side here is a plot where we asked the question about, in our 4 categories of melters, what about protein length? Because it had been suggested that the bigger proteins are more thermal labile. And statistically, we can see that. I mean, if you go from the black to the red to the gray to the blue distribution here, you can see that that the average sort of length gets higher and higher, and it has some ridiculous p-value. However, really, when you see how broad these distributions are, you can't really conclude from that, oh, if a protein is big, it will be thermally not very stable. Now, we can take a little more refined look at this because when we now look at all of the species in this plot in the middle, and again, now this time map out the length of the protein in terms of numbers of amino acids and plot it against the mean melting point of each species, then actually we do see that while the eukaryotes are all over the place, the bacterial species, they do at least follow some trend. And I acknowledge that the statisticians are going to get shivers when you make correlation plots that only have 6 data points in them, but there is some correlation. And there's actually some biology behind this because bacteria have very few ways of making complex functional proteins other than making them bigger. And it seems that Antarctica and Subtilis are a little better at doing this, or if you turn that around, is that if you want to live at very high temperatures, you have to live with the fact that you need to make shorter proteins.
A couple of questions, could we do that? What was the difference in the heat tolerance among homologous proteins between Thermophilus and E. coli?
Yeah, good, very good question. Gigantic. It's pretty much the same distance than for all others. So if we look at the very same protein, or it's actually not as easy to trace all the way from Thermophilus to Antarctica what the same protein is, but we managed to do this for a couple of 100 cases, and we saw the same thing. So even a functionally homologous protein is vastly different in thermal behavior, but if you are that distant in evolution, actually also the identity level on the amino acid level is also not very high anymore, even as little as 20%. So you can conserve a certain protein function, even though the level of amino acid identity may be as little as 20%.
Did you observe a correlation between half-life of proteins versus thermal stability?
Coming in a second.
And one more. Were there more cysteines in the Thermophilus proteins as compared to the E. coli proteins to explain the increase in thermal stability?
There were actually less cysteines, because this beast lives in thermal vent and then is such a reducing environment that building cysteine bridges won't work. Too much reducing power around.
Does any particular amino acid contribute to high stability?
We'll come to that in the next slide.
And maybe the last question in this batch. In the experiment where you mixed lysate from E. coli and Thermophilus, the figure that you showed, was it for organism-specific proteins? Were there any common proteins between those organisms?
That I can't answer for sure, but I would assume that there are functionally common proteins in there, because our chance of detecting something is if they're more highly abundant, and so we're assuming that they are more like housekeeping proteins, but I would have to look that up.
Okay, thank you.
Okay, on the right-hand side, just to finish this, is we can also see some trend in protein abundance, that the higher abundant a protein is, the higher the melting point it also is. Again, statistically superbly significant, but if you look at the boxes and whiskers, I'd say they have very little predictive power in protein abundance too. That may still mean something, but it's not easy to interpret. Now, okay, so as I said, that we looked at a lot of things that turned out to be very frustrating. And in the supplement of that paper, you can actually look at these frequency plots that I show here for every single amino acid in the alphabet of proteins. And we actually mostly saw nothing on an individual amino acid basis. However, when we group some amino acids together, like polar amino acids, particularly those 5 here, we actually did see that those thermophiles had a lot less of these polar amino acids compared to a mesophile like E. coli. And when we then turn this sort of around and ask the similar question, what about the hydrophobic amino acids then? The difference isn't quite as pronounced, but it's quite clear that Thermophilus makes more use of hydrophobic amino acids. And that the interpretation of that is in this heated environment, you don't want too many of those polar amino acids around, because the water shells and so can actually disrupt the structure of your proteins. Whereas if you're using more hydrophobic amino acids, you're generating a more compact structure, which goes along potentially then with higher thermal stability. But on an individual amino acid level, we actually saw virtually nothing. And also, the same when we looked at secondary structure elements like alpha helices or beta strands, and here are all of our species, you don't really notice a profound difference between them. So, and if we then go on and more specifically ask the question, what about disordered structure then? One thing that we did see is that the higher organisms all have a lot more disordered structure than the bacteria. Well, that is something that people had known long before. But when we then again look at our 4 categories, in this case in human, we find if we plot that again along the axis of percentage of disordered structure, we essentially saw some continuous and not very telling characteristics between these 3. But the non-melters were interesting, and we can't say that we understand it, but the non-melters essentially show this bimodal distribution, where you have a lot of proteins that don't melt, but they actually have not at all any disordered structure. But then there are these disordered structure proteins with 100% of it having no structure, and they also don't melt, and then anything in between. So it's hard for us to really grasp what this is, but we are just observing that this is the case. And here I showed you these 2 examples before. Both proteins are almost completely disordered. One of them doesn't crash out of solution, the other crashes out of solution rather quickly. So at this point, what we're concluding from this, and that may be not particularly satisfying, but we really looked near and far for physicochemical properties of peptides, amino acids, proteins, secondary structure, what have you. We think what nature actually has done is to optimize a protein sequence, almost like a protein design and evolution person would do, is randomize or do biotechnology on every residue until you hit the properties that you want. So it's well known that you can engineer enzymes to be thermostable by exchanging many or certain amino acids. And then you can use these enzymes, let's say in washing powder very easily and still heat up your washing machine to 95 degrees and those proteases may still work. So that's currently the interpretation, that it's careful sort of protein optimization for the purpose of making them more thermostable if they need to. Now, one snippet that we thought was interesting is when we go back to humans, we observed that there actually is a difference in the thermal stability of proteins depending on where they are. And particularly, we found that proteins in the respiratory chain seem to be rather more thermal stable than other proteins. And we actually even did a functional experiment along this line, because there was a report in the literature that argued that mitochondria might actually operate at close to 50 degrees in a cell. And that we found surprising, but here at least we saw that, well, mitochondrial proteins are a little more stable, but particularly so the respiratory chain. This is actually where heat is generated, because in the mitochondria, in the respiratory chain, protons are being pumped across the mitochondrial membrane. And there's a lot of what is called proton leakage, where the protons flow back without propelling the ATPase, ATP synthase, and that annihilation of proton gradient generate heats. And so together with some mitochondrial experts here on campus, we actually did we measured oxygen consumption by mitochondria, either in intact cells or in permeabilized cells. And this is the gray and the yellow bars. And we actually found that in the permeabilized cells where glycolysis, where these mitochondria are basically independent of glycolysis, because we made holes into those cells and basically fed them with substrates for the respiratory chain, we could actually see that they would hold on to functionally using those metabolites at higher temperatures than the intact cells. Whether that means mitochondria can actually operate at 50 degrees, we don't know quite, but at least very locally in the respiratory chain, maybe these proteins are more thermal stable simply because they have to be, because the environment is warmer. I'll skip over that in the interest of being able to look at a second example. Olga, if we still have time, and I already apologize that even 45 slides are not are far too many for 1.5 hours, but
we have 10 to 15 minutes.
We have 10 to 15 minutes. Okay, good. The second story that we published a while ago is about protein turnover and also proteome form turnover that more and more turns out to be an important regulator of cellular homeostasis, and therefore people have begun to call also these proteostasis. So there are important elements of regulating the lifetime of a protein, whether you want to look at and make protein complexes or protect yourselves from misfolded or toxic proteins. This interplays with signaling. People have found that there are phosphorylation signatures on proteins that mark them for degradation. A lot of diseases are these days referred to as proteostasis or dysregulated processes like Alzheimer's disease, Parkinson's, and what have you. But also in drug research, this is becoming more and more interesting, either because these drugs interfere with proteostasis proteins themselves, like proteasome inhibitors or inhibitors of the HSP90 machinery, but also all of those covalent irreversible inhibitors that are more and more entering drug research, because once they are irreversibly inactivated, then of course the action of the drug will depend on the turnover of the time essentially when the cell will replenish that protein. So we've used TMT actually now in conjunction with SILAC labeling in what looks like a rather complex experiment, but was also sort of a data analysis nightmare for Jana Zecher, who ran that project. But it turned out to be extremely interesting to do. So we wanted to measure this protein half-life or turnover on a larger scale. And again, we are not the only ones who've done these experiments, but the twist of TMT came in extremely handy. So the classical way of measuring half-life of proteins would be a pulse chase experiment, and we did this with Silac, where you started with light labeled cells, and at some point in time you switch them to heavy, such that then all the new proteins that are made from that point forward would incorporate the heavy isotopes, and the red proteins would sort of be the old stuff that's still there. And then we basically collected samples along this time frame of the beginning to an infinite number of hours, which I think was about 5 days. And then each of those time points were encoded by TMT labels, and we then mixed them all together and then did some fractionation and then LCMS analysis on this. And it turned out by the times we chose that we usually got Silac pairs that were roughly 1-to-1, because it depends, of course, on the doubling time of those cells, what that exact time frame here is. But then we asked the mass spectrometer to both fragment the old proteins and the new proteins, and that from MS2 spectra we would then pick a couple of those fragment ions to do an MS3 experiment where we would detect the TMT reporters. And that enables something rather interesting, because now you can plot those TMT intensities of the old proteins over time, and you can fit curves, degradation curves to them that tell you how quickly those proteins disappear from the cells because of degradation. And conversely, the blue channels, or the newly made proteins, we could now generate synthesis rate curves for each of those. And that is important to use TMT in an MS3 mode, because if you want to measure synthesis, really your time zero must have 0 value for that peptide. And if you only do this in MS2, I can tell you from experience that most of the curves start at 0.2 or even 0.3. So then when you fit these curves, you're actually getting quite wrong data. So here the combination of Silac and TMT allowed us to get these degradation and synthesis curves in the same experiment. And from that, essentially for each protein, ignore the stuff on the left-hand side. There's just a lot of quality control. But that allowed us to generate synthesis and degradation curves for a lot of proteins. And you see 4 lines here, which are 4 replicates, and 4 lines here are the same 4 replicates. And of course, the crossing point here always would tell you something about when you have 50-50. But then you have to correct this for the doubling time of those cells. And then you can compute, for example, for this cyclin protein here, that it has a half-life of about 3 hours, whereas this ribosomal protein has a half-life of about 100 hours. And when you plot that out for all of the proteins that we've got, then you can see that on average, the half-life of these proteins is about 1.5 days. But then there are also some that have really short-lived and others that are very, very long-lived. That to some degree correlates with the copy numbers per cell. So the more abundant the protein is, the longer it tends to live. That makes, of course, some sense. Again, when we ask the question, do longer proteins, are they less stable or have they shorter half-lives? The answer is no. And again, when we turn to more structural elements, we saw actually that the polar amino acid and coiled-like structure had a destabilizing effect, and then hydrophobic amino acid had a rather stabilizing effect, akin to what we actually saw in the temperature experiments in my first study case. Okay, we also broke this down into parts of the cell, or what they do. Here in parts of the cell, you can and just rank them by their half-lives. This is now in log 10 scale. Here being on the left-hand side being the most unstable ones, on the right-hand side the most stable ones. Again, very hard to make general statements about it. You can see perhaps a trend along this if you want to order it this way, but it's very hard to really conclude on a protein by protein basis anything from where they are in the cell. What was more telling is actually an enrichment analysis of the half-lives, where we saw that many people already saw before, is that particularly transcription factors tend to be short-lived, which of course makes sense for the cell if it has to respond to some changing situation. It either wants to make more proteins quickly, or it wants to stop making certain proteins quickly, and the transcription factors would be one way of doing this. Whereas the more stable proteins also tend to be more enriched into processes that the cells need all the time. Again, here coming back to the respiratory chain, we also again saw something that we already saw with the thermal stability. We looked at which proteins would be actually more stable or less stable, and we actually found that proteins from the complex 1 of the respiratory chain tended to be rather unstable, and we wondered if that is because of oxidative damage. And if we look at here the respiratory chain over here, in complex 1, this is where also a lot of oxidative stress actually happens, and the half-life of the proteins in complex 1, at least on average, were, this is again a log10 scale, quite a bit shorter than that of others. And basically the point of this plot down here is when we map this out onto the structures of these proteins, we see it really only affects proteins that are in the lumen and not the proteins that are in the membrane. And one potential explanation for the lower or higher turnover of these subunits is that they potentially accumulate oxidative damage and therefore potentially lose their function, so they have to be exchanged more often than those proteins that sort of sit in the membrane, are not exposed to as much oxidative stress. So in the last 5 minutes, I would like to quickly go through, again, coming back to something computational, if I may, where TMT is also beginning to play a role. And some of you may have heard about our Proteome Tools project, where we've some time ago started making a lot of synthetic peptides with the aim to represent them as a human proteome in terms of synthetic peptides, and essentially to create sort of a ground truth data set for LC-MS-based proteomics, and then build prediction tools from the tandem mass spectra that might work for any protein from any organism. And that kept us busy for some time, but it was and actually is a very fun project where we made all these peptides and fragmented them in a lot of ways and got hundreds of millions of PSMs, and the data is all there for people to use. And initially we did this all label free, but we've then added a TMT component to this to make, for example, spectral libraries that are available from various places. And just a few snippets for why we actually wanted to do this. Much of it was driven by the wish to have better error control in proteomic studies other than using classical target decoy type of error control methodology, because we saw that they all have their limitations and that can accumulate in very large data sets. So it's quite clear why this may happen. For example, you have here a peptide or an experimental spectrum here in point of the signals pointing up that Andromeda matched to a sequence like this. And then the second best match to that very same spectrum is a peptide with a rather different sequence, and the score is almost the same. It's hard to distinguish for the search engine because the search engine in this case kind of doesn't really know what intensities to expect from those peaks. Whereas when you make synthetic peptides for exactly this peptide and record their spectra, you can see from a mile away that this sequence, this synthetic peptide spectrum, looks almost identical to the one that Andromeda has matched as a second best, and the best one actually is something completely different. And so we can clearly rule out this one and say this is the much better match. And we wanted to do this systematically, and because there was a lot of data there, Sigi Gesulat trained a neural network on this data to basically try to predict the fragment ion intensities and also the retention times of all these peptides. That turned out to work extremely well, and that made us extremely happy, because at the beginning it took us a lot of time to find a funding agency that would give us some money, because everybody said, why do you want to do something as boring as make a lot of synthetic peptides? And we are very happy that we did, because it turned out that this prediction tool works rather well. And in this plot here, it may be a little complicated to understand at first, but essentially what it shows you is a spectrum of a peptide of this sequence. And in red are the Y ions and in blue are the B ions. And if you see a lot of red and blue, that means that the actual experimental spectrum and the prediction made by that machine learning tool agreed extremely well. And if there was a little bit of green, then there was a little more signal intensity in the experimental spectrum. And if there was more yellow, then the prediction would have thought there should be a little more intensity here, but we didn't see that. But when we looked across many of those spectra, we saw that this tool that we built that we call ProSit did rather well when we compare experimental spectrum to predictions. So in Spearman in Pearson correlation world, that would be north of 0.99. In spectral angle world, it's still better than 0.9. And that has enabled or is enabling quite a few things. One is, for example, rescoring data where large search spaces are a problem. And one example is here shown for metaproteomics, but it would also be true for HLA peptides or for doing proteogenomics, where you have to consider a lot of sequences, and therefore your error control becomes harder and harder just because your sequence database against which you're searching becomes bigger and bigger. And I would like you just to look at this part of the figure. When we search a metaproteomic data set that also contains human data against just the human database, then we'll, let's say in this case, find about 3,000 peptides. If we make the database bigger by asking, well, let's search against the human sequences of UniProt and all bacteria, we all of a sudden begin losing identifications we had before. And we also gain a lot of identifications of bacterial peptides that we didn't have before. But then when we make the search space even bigger, which would be that huge green blob down here, Andromeda really finds it very hard to find anything. Now, if we then basically ask PROSIT to predict all these spectra and ask now the question, how well do the experimental and the predicted spectra match to each other, we can actually see that at 1% false discovery rate, we all of a sudden get 50,000 bacterial peptides. And yes, we're also losing human peptides, but not quite as many. And we can also crank this FDR up to 0.1% without really taking a big hit. So we think that all these spectra are very useful for this type of rescoring exercises. And the reason why this actually works is, that it's better able to discriminate false and true matches. Over the past few months, we have also sort of extended this to non-tryptic peptides or modified peptides, and it seems to be working quite well because a lot of these peptides that we've made are also modified. And here's an example where phosphorylation site localization may be improved. And I'm just going over this reasonably quickly because I wanted to come to the TMT part, which is sort of the latest addition. So we've also now trained PROSET on TMT labeled peptides. And you can see these mirror spectra up here where we have this particular peptide that is TMT labeled, and the peaks pointing down are the actual experimental spectrum, and the peaks pointing up is the prediction. And again, and comparing and contrasting that to the non-TMT labeled version of the same peptide, which actually does look different to this spectrum. But when we look at the violin plots, the story is the same for as for the typical unmodified peptides and non-labeled peptides, that it seems that PROSIT is learning what these spectra should look like. And therefore, once we are able to release this, we hope that people find good use for this. And with this, really, I'm coming to an end of the presentation. I apologize for taking a lot of time, and I hope I didn't bulldoze over too many of you with too much information. But of course, we have now some more time for questions, I guess. And I would like you also to just take a moment, if you have, to look at Proteomics DB, where many of the things I talked about eventually end up in, either be it large data sets or be it computational tools. And Proteomics DB can either be explored with your browser or with APIs for the computationally inclined people. And we hope that this continues to serve the community well. And with that, I really have to say thank you to a lot of people that have done all the work, and also a lot of collaborators around what I presented today, around be it the Meltome Atlas or be it creating Proteomics DB or creating proteome tools and PROSIT. So with that, I say thank you for all of you who might still be here. I have no idea how many of you are still here because I don't see how many people are in the presentation, but it doesn't matter. So thank you for all of you who stayed until the end. And I'm happy to take more questions.
Thank you so very much. We have a round of virtual applause from over 450 participants who were listening to your presentation. So we have time, we have questions coming up more. So let me just ask one question, which kind of maybe wraps this up from one of the participants. So what is your current decision tree for proteomics experiments? So how do you decide when you go with LFQ versus TMT?
Yeah, so we really very carefully define the set of questions we want to answer right at the beginning. And to the extent possible, we're going to make that decision rational. Sometimes it has to be practical. For focused small experiments, we actually tend to do more and more and more TMT because it allows us to put replicates together and directly compare experimental conditions. And we have taken the decision in the lab that whatever we would do with TMT, it's always going to be MS3 because we want to capture some of that dynamic range and accuracy that we can only get from MS3. We know we get less data. We get fewer peptides, fewer proteins, but the quality of that quantification is much better. So every time we do TMT, in the end, it's always going to be MS3. Most of the sort of largest experiment, let's say for the melting experiment, there was really no other choice. You can't do that many experiments in a label-free fashion because it would be 10 times more mass spec time. It's just completely impossible. And it's not that my lab has 20 instruments. No, it's not even that my lab has 10 instruments. So we have to also be mindful of these practical issues. But I think the short answer is we really try to operate a fit for purpose approach every time we come up with a study or project.
Well, thank you so very much. So we have a lot more questions. So I'm carrying the questions over to the Google Doc. And Bernhard, if you don't mind, take a look at the Google Doc and see if you can give answers. There are many questions, so to the extent of your ability, just answer a little bit. And maybe let me also mention two things. So tomorrow and Friday, we have 2 days dedicated to work with Skyline. And it is my understanding that Brandon, who is the developer of Skyline and the leader of the 2 sessions, will be talking about integrating Prozit with Skyline. So
yes,
this
I forgot that. That was actually a fun summer intern project that we got Prozit into Skyline. So I'm very happy about that. Yeah.
And another plugin is that next week, I think it is Thursday next week, we will be talking more about statistical analysis and we will have a presentation dedicated to MSStats TMT. So there were many questions about normalization, reference channels, things like that. So we will certainly be discussing this in the context of MSStats.
Okay.
So thank you, Bernhard, once again. So it is wonderful to hear from you. And I think it will be a very, very useful resource for many people who watched now, but also who haven't had a chance to join us. So it will be available on YouTube. So you can share this with your group members. You can share this with your colleagues. So we're a little bit over time. So let us take a 10-minute break so that we can regroup a little bit and stretch our legs. So some new people may be joining us for the second part. So we are staying on the same link, so you don't need to go away. So we'll just take a break for a few minutes and we will come back at 12:45 Eastern. Okay. Thank you so much. And thank you again, Merham.
Hey, Olga. Hey, Mina.
Yes.
I'm on.
Yes.
And did you get my slides?
Yes, I got it. And then the slide is available. So participants or attendees can get your slide, but could you try to open it?
Yeah, I just share screen.
And just so you know, you're broadcast to everybody.
Yeah.
Perfect. Thank you so much. So we'll just wait a few minutes and we will start in 5 minutes.
Yeah. Right. It works.
Terrific.
Yeah, perfect.
Okay. Good. Hey, Mina.
Yes, yes.
So you can hear me. I wasn't sure whether I was disconnected,
so
but it's still on.
we will go ahead and get started now. So let us go ahead and get started with the second part of the program. So those of you who are joining us just for this second part, we welcome you to this third day of the May Institute. We are very excited to have an opportunity for us to come together and discuss topics in mass spectrometry and proteomics and computation and quantitative studies, and still, be productive and have fun despite all the circumstances. So I hope that you will find the program in these 2 weeks useful. So if you are joining us for the first time in terms of logistics, just so you know that at the bottom of your screen, you have two boxes. There is a chat box and a Q&A box. So the chat box is best to contact the panelists for some logistical issues if you're missing a URL or you need some particular kind of logistics question. But if you have questions to the presenter regarding the technical topics, so please use the Q&A box, and it allows us to organize the questions. And so we will interrupt the speaker as possible and voice your questions as much as it's possible. And if we have more questions than what we can actually ask, we will carry them over to the Google Doc. So we have a Google Doc which has links to the slides and also additional questions. And then if the speaker has a chance to answer, they will answer that. So this is how it will work. And so now let me introduce our second keynote speaker of the day, Professor Rudi Ebersold. It is my particular pleasure to welcome Rudi back to the May Institute. Rudi has been a participant with us for a number of years, and we had together co-organized and co-led multiple short courses and multiple events like that. So it is a very special honor for me to introduce Rudi, although I'm sure you know that Rudi doesn't need much introduction. So Rudi holds a PhD in biology from University of Basel, and then he was a postdoctoral fellow in Caltech. He had faculty appointments in University of British Columbia and University of Washington. In 2000, he co-founded together with Lee Hood and Alan Underem Institute for Systems Biology in Seattle, which was a pioneer in developing interdisciplinary approaches to biological investigation. And then in 2005, Rudi went back to Switzerland to become a founding director of the Institute for Molecular Systems Biology in ETH Zurich. And Rudi made numerous contributions to our field, and this includes approaches such as ICAT to labeling, quantitative mass spectrometry. Rudi's lab made important contributions on the computational side, such as transproteomics pipeline and databases such as Peptide Atlas. Rudi made really important contributions to targeted proteomics and SRM, and also more recently to data-independent acquisition and SWAS. And what I personally find particularly fascinating in Rudi's work is, on one hand, the connection between the technology and the biological applications, and how with the technologies that are developed in his lab, Rudi makes sure to show their practical utility for biological studies. And then the second thing, which I also find particularly amazing for Rudi's work, is his mentoring and his way of interacting with junior scientists, and how many people from his lab went on to become very influential independent investigators. So it is really my honor to welcome Rudi back, and we very much look forward to your talk, Rudi. Thank you.
Okay, thank you very much, Olga. It's fun to be here in my office and give this talk, and hopefully we'll have some discussion. It would be, of course, much nicer to be in person in one site, that one also will have personal interaction, but it is not possible right now. And I would like to acknowledge and also congratulate Olga and her team Mina, Ting, and Kylie, and the whole team, that they managed to put together this May Institute in this entirely new format. I think it is working quite well. And the benefit is that instead of the number of people that can be reached is of course larger, but of course the interaction is much less personal. So I hope we will still have a good exchange here in this format. And now I would like to start. So Olga created a title, Modern Technology for Modern Biology. It's a good title. I changed slightly the focus. I mean, it's not the focus, it's the same focus, but what I would like to focus on in this hour or so is how we can use the data that we create by these now pretty amazing proteomic techniques to learn new biology. Because ultimately, it is great to have mass spectrometers. They are very powerful. They produce beautiful data today. But ultimately, I think it is about what does this data mean, and what can we learn about biological processes that we do not know yet. So this is basically my topic. So let me start out by saying that biology and also medicine is essentially about function and phenotype. So we see here on the right-hand side two individuals of the same species, and everyone will immediately see that they differ in one phenotype, of course also in others, but one is size. So everyone can observe that. And of course, for a long time, one did not understand why is this, that some people in the same species are so different from each other. And in medicine, of course, the same questions arise. A person may be afflicted with a particular disease, another person not. And so we would like to know what is the underlying difference between the molecular difference between these phenotypes altered, like disease-associated normal, so that medicine could eventually undertake something to reset this disease phenotype. So we have been conditioned in the age of molecular biology over the last few decades that we use molecules as surrogates for function or phenotype. So we believe, I think this is one of the bedrock assumptions in molecular biology, that it is the molecular constellation of a cell or an organism that determines its phenotype. Of course, we don't know the rules, at least not precisely how specific molecules affect a phenotype. There is very clear indications of, for instance, in Mendelian genetics, where the link between the molecular landscape of the genome and the phenotype is quite clear, but usually it is much more complicated. And so as proteomics researchers who operate in this field of molecular biology, we would assume, and I think this is one of our convictions, that the better or the best maybe surrogates for function is proteins. And why is this? Because proteins, of course, carry out most of the biological functions. They catalyze them and also control biological function to a large extent. So this has led to the notion that if we would understand and could measure and understand the function of essentially all proteins that are in our system, that is usually a cell, or could be also a multicellular organism or an organ, then we would make a lot of headway with understanding the biological processes. This is an assumption, and I think this is the guiding assumption that many biologists and proteomics researchers pursue. So here is just a few numbers to illustrate how complex the system is, that we nicely can draw as a cell, and we can give it a nucleus, indicate a nucleus and some other structures. But in the reality, this is an exceedingly complicated and actually amazing structure that we are studying here. So it is a highly complex, multiplexed chemical reactor. At any given time, hundreds, if not thousands, of biochemical reactions are concurrently occurring. the size is very small. We can see them in a light microscope, of course, these cells, and the volume is approximately 1,000 femtoliters. Size is micrometer. So with the naked eye, we don't really see the cell. There's exceptions like oocytes, particularly frog oocytes, but generally these cells are in micrometer range. They contain about 8 billion molecules, protein molecules. Of course, not all are different. There's groups of proteins which are from the same gene, which are inherently quite homogeneous. But the number of molecules is very large, roughly the same as number of people on Earth. And the protein concentration in one of these cells is approximately 300 milligram per milliliter. So this is about maybe 2 orders of magnitude more concentrated than what we usually do when we do biochemical experiments in vitro let's say, in vitro kinase assay, where we usually operate in the 1 to few milligram per milliliter. So the conclusions here is we are dealing with a very complex system, and that consists of a very large number of molecules, and they are organized in a soup or in an environment that we really cannot achieve or recapitulate by in vitro biology, by biochemistry. So if we want to analyze these proteins based on what I just said, then we can see what the challenge is. And now I would like to make a few comments on how amazingly fast this proteomics field has developed. So I go back a few decades. This was the age of where really protein identification by their sequence became feasible. But this was roughly in the 80s. That's when I did my PhD thesis. And at the time, this is what I actually was doing then, is to sequence proteins. And these proteins were proteins that had to be highly purified. And the type of proteins I worked with were relatively short polypeptide chains, particularly amino acid sequences of immune globulins, where the light chains have approximately 150 amino acids. And to sequence one of those with a tedious stepwise degradation of one amino acid at a time took me about 6 months of work. So this translated in about, at the time, in approximately 1 amino acid finished sequence per day. And to do that, I needed about 10 milligrams of pure protein. So most of the work actually went into purifying this amount of protein. And this is, of course, those who work in protein biochemistry know that to purify out of a biological sample milligram amounts of pure protein is a sizable undertaking. So let's move forward a few decades to approximately now. We can claim, I think this would be not contested numbers would not be contested by many, that we can identify approximately 10,000 proteins per hour in a modern instrument. This translates in about 1.5 million amino acids sequenced per hour compared to 1 a day some decades ago. And the average amount of protein required from a Wi-Fi sequence of proteins and complex mixtures would be about 250 femtograms. This is enormous progress. And in genomics, there's a famous picture from the National Institute of Health of Genomic Sciences, and which shows that the progress in genomic sequencing in terms of sample throughput and price has been roughly following Moore's law, actually exceeded Moore's law. So it's faster than what Gordon Moore predicted for the duplication rate of microchips, basically electronic devices. So here we see, I made some assumptions here, and the student in the lab, Amir Banai, made this plot. And based on the results that I just showed in the previous slide and some number of milestones in the development of mass spectrometric methods out of the protein chemistry methods that were used some decades ago, we can conclude that in terms of the required protein amount to sequence a protein, the progress has been faster than Moore's law. And also in terms of throughput of proteins measured, for instance, by proteins or amino acids identified per time unit, we also see that it is faster, that exceeds Moore's law which I think is an indication how rapidly the field has developed. So not only have we sequenced a lot of proteins, there's also a lot of resources that allow the field of proteomics to really do the analysis more reliable and really generate data with high quality. So I want to just now in the following point out a few of these resources which are generally accessible, and then move on with what I really would like to talk about. So one resource is the Human Peptide Atlas, and it shows that it's a part of the of a consortium to acquire and share the data that have been generated by these mass spectrometric studies in many laboratories. And this was started in 2004 by visiting scientists at the time, Frank Desier, together with Eric Deutsch and Alexei Nesvitsky at the Institute of Systems Biology in Seattle. So we thought we would collect data that people generate anyway, and once they publish a paper, we would ask them to donate the spectra. And then Eric Deutsch particularly developed a computational pipeline to conclude which peptides and proteins were represented by this massive amount of data consisting of hundreds or thousands of data sets. So this is roughly a state of today. For a few months ago, so hundreds of millions of peptide spectrum matches have been established from this very, very large number of spectra that have been donated from thousands of experiments. There's about 1.8 million distinct peptides that have been identified, and there is for vast majority of the proteins in the human proteome, like for 82%, there is unique identifier peptides so canonical peptides, which identify the protein distinctively. And for about 14, there's evidence, but through the redundancy of peptides, we cannot really say whether a specific protein is in there or whether it's a member of a family. It's usually referred to as protein groups. So this means that the community of scientists who have donated the data, which is also, for instance, accessible. The same data is also accessible through PRIDE in the consortium called Proteome Exchange Consortium. Then this means that we basically are able to cover most of the proteins that are in a human species, but the same applies to many other species. So this is, of course, a lot of work to create this coverage, and it will be. And we also wanted to, at some point, see that through spectra would not only be accumulated to say we have this protein has been identified, but that we could use spectra in targeted experiments to then remeasure and confirm the presence of proteins in a hypothesis-driven way in samples. So Ulrike Kusebauch was the main driver of this project, which was done between our group and Rob Moritz's group at the ISB. And she undertook to synthesize a large number, about 150,000 peptides, which were selected to represent most of the proteins of the human proteome, and to generate spectra of these peptides in different mass spectrometers under different fragmentation conditions. So we call this the Human SRM Atlas, and it's a resource for targeted assays to quantify essentially any protein in the human proteome. This effort now is continued and exceeded, in fact, and massively extended into phosphopeptides by Bernhard Küster, who just was on this session before. And I think this is a great resource that Ulrike created and now is being extended by the work of Bernhard Küster and his group. And I think these reference spectra are really a useful resource to do reliable remeasurements by a variety of targeting techniques like PRM, SRM, and also increasingly DIA swath measurements for the measurement through whole cohorts of samples. So I don't want to go into the statistics here, but for most peptides, for most proteins, there are 18,000 proteins of the human proteome. 5 or more peptides are available that are, in fact, mapping to these specific proteins. There's only 22 proteins for which there is no peptides available to identify these proteins in a sample by targeted mass spectrometry. So these are resources to say how was the protein covered, or what coverage has been achieved, and resources to do measurement on any specific sample with to test the hypothesis whether a protein is present and in that sample, and to quantify that protein. But as we will see later on, the greatest benefit of proteomics these days is probably not to say, well, we have identified a specific protein, but to identify a relatively large number of proteins, as many as possible, in as many replicates as possible. And so the idea of measuring proteins in sample cohorts with high reproducibility and quantitative accuracy has been, of course, in the forefront of technology development. And our group has worked intensely to bring along these techniques that allows us to measure reproducibly proteins over hundreds of samples. And the solution that we came up with is data-independent analysis, initially implemented with the SWOS technique, which will probably further be discussed in this May Institute. And we now, and I want to show just one slide to show that even these DIA techniques and the measurements are also really progressing very fast as a technique to acquire data across sample cohorts. So this is from a collaboration that we have undertaken with the group of Matthias Mann and the group of Hannes Röst in Toronto, and also supported by Brucker, who developed an instrument which is called PASEF. So this is basically an instrument that combines a quadrupole TOF instrument with a ion mobility device that stacks up ions so that the ions can be accumulated before they're being analyzed. And so this allows us to measure, basically to make much better use of the ion statistics. So virtually, well, I shouldn't say virtually all, but most of the ions that are being created at the source are actually used to create fragment ion spectra. I don't have time here to get into this technique. There's an article in bioRxiv that describes the instrument as well as the technique. I just want to show that this machine, in conjunction with this with the DIA technique, really provides quite fascinating data. So this is a replicate analysis of 200 nanograms of total lysate of HeLa. So this is a relatively small amount that is injected on a 100-minute gradient. And the system identifies quite reproducibly about 65,000 peptides, corresponding to about 7,500 proteins. And I should say that these proteins are inferred from prototypic peptides only. There's no protein groups in there. And when we look what is detected in a single shot from this analysis, it is the blue part of the curve here, and it's the proportion of the white curve, which is the library that is which was generated by 24 reverse phase fractions of separated fractions, and by DDA analysis of the same instrument. So then there is the reproducibility. So most peptides are covered in the repeats, except some where the signal to noise becomes very unfavorable. And there, of course, some peptides are only detected in a subset of the replicates. So this is just to show that the technique of acquiring data from large cohorts to generate data sets which are of quite deep into the proteome and are quite quickly generated has also been progressing. And I think this roughly represents now the state of the art. And it's also a data point from this Moore's law calculation that I showed before. It's one of the last data points. So we would conclude here that for many biological explorations where we would like to learn new biology, we will need to have a first data set. And we used to think about identifying as many proteins as possible from a sample. I think this is still valuable for some applications, but for many more applications, it is more valuable to have a data matrix where we have N samples and P proteins. And this data matrix can then be used for clustering, for correlation type of analysis. And I will then expand in the next minutes on how we use data created by this type of analysis, this data matrix type, to learn new biological insights in a number of different scenarios. So just a couple of comments on this matrix and its shape and dimensions. So we learned, this is actually from a collaboration we have with Olga Witek, and especially with a postdoc in her group, a student in her group, Ting, that Ting simulated that if we have the choice to make this matrix broader, that means have larger numbers of samples, then we generally create more useful information than if we go deeper into the proteome. Of course, we would like to do both. We would like to go broad, and we would like to go deep. But there's always a trade-off with resources. And if we would have the choice to allocate certain amount of resources either into a larger number of replicates, larger number of samples, or to use the same resource to dig deeper into the proteome of fewer samples, the simulations that clearly show that it is a benefit to go for more samples, potentially at the cost of a larger number of proteins. But of course, we would like to go deep as well. And this DIA technique that I just showed allows us to go quite deep, quite fast, and also to go through a large number of samples. So all the subsequent analysis that I'm going to show, we now discuss how we can extract biological information, are always dependent on data matrices of some sort where we have a certain number of samples that are measured in a certain number of proteins. And from this data matrix, as opposed to a list of proteins, we try to conclude new biological insights. So to summarize this first part, what I would like to try to illustrate, just as a setting the scene for the subsequent part, I try to justify why proteomic researchers focus on proteins as their subject matter of study. That is because proteins, we think, are very close to biological function, because they catalyze and control most functions. Proteomics is justified by the focus, therefore, on the function modules or molecules of the cells. I try to show that proteomics has made enormous progress over the last few years. There's now solid evidence for most proteins in human species and in other species. There's reliable spectral assays for virtually the whole predicted human proteome that can be used for hypothesis testing using targeted analysis. Reproducible, deep, and quite accurate measurements of proteins in single-shot DIA methods is certainly possible. And Bernhard talked about TMT, which has, of course, measurements which has very similar objectives. And these techniques allow us to generate extended data matrices of actually quite good quality. So we will conclude here so far that we can measure proteins amazingly well, but how can we translate these large-scale molecular data into functional biological knowledge? So this is now the points I would like to discuss in the following. So as a guide through these next few kind of vignettes, I use a graph that was generated by Isabel Bludau, who was a student in our group till a few months ago. And she wrote at the end of her thesis a review where she discussed the complications of the proteome and the challenges it poses. So we have now said so far in this presentation that we focus on the proteome, and most proteomic studies are focused on an instance of the proteome that basically consists of any one of a number of transcripts or translation products of transcripts from any one of the genes. So we know, of course, we have roughly 20,000 protein coding genes in the human proteome, and these are translated. And when we identify proteins, we would typically say we have identified a protein from this gene, but we do not make a specification typically which form of that protein we have identified. And we know, of course, that there's a massive expansion of the number of molecules which we believe are functional molecules that are generated from these 20,000 or so protein coding genes as we go along the axis of gene expression. So this graph shows that we see that, of course, these genes are transcribed at first, and then at the level of transcription, there is a diversification, for instance, through alternative splicing. There is then the further amplification of the number of potential products that these alternatively spliced transcripts can be translated, and in different forms, for instance, through alternative start sites. And then, of course, there's enormous diversification at the level of post-translation modifications. And then there's a further enormous combinatorial space where proteins, or more specifically proteoforms, derived from these amplification or expansion mechanisms can, to some extent, combinatorially form protein complexes. And at the end here, we have the phenotypes. And what we would like to know, of course, which functional units, which proteoforms, which protein modules, or combinations thereof are actually functionally and causally causing a particular molecular function and potentially phenotype. So this poses enormous challenges, and because we can measure these things quite well, we can measure thousands of PTMs, for instance, phosphosites or UB sites, ubiquitination sites. We can measure different proteoforms. We can measure number of protein complexes, but we don't necessarily know what they functionally mean for the cell and the determination of phenotypes. So this is the topic I would like to discuss, approaches where we can make basically determine, based on proteomic data, the functional significance of the proteome. And I will do this at 4 different stages here as we go along the axis of gene expression. We will talk about the functional insights into translation products of a particular gene. This is what most people measure. We'll then go to some functionalization of PTMs, functionalization of complexes, and then functionalization of proteoforms in the context of specific complexes, namely how proteoforms associated with complexes. So see how far we get, because this is quite a lot of material and we'll want to be cognizant of the time, but this was what I thought we would do. So let's go to the first question. And the first level, what is the functional significance of the proteome at the level that most proteomic laboratories operate, including ours, where we say we have identified with a mass spectrometer specific proteins. We may not be able to exactly distinguish the proteoforms, but we define proteins basically as translation products of a specific gene, without usually differentiating between these specific products that, for instance, could be generated by alternative splicing or by PTMs. So we can, of course, do direct biochemical analysis of the proteins. And this can be done and has been done very successfully by in vitro reaction. So an example here is a direct experimental determination of a biochemical function of a protein in the case of a protein kinase. So we would have, this is just an example, there's many other examples one could use, but we would have a protein that is a substrate of a particular protein kinase. One might use a panel of proteins if one does not know the substrate specificity of this kinase, and would simply incubate this target protein with, for instance, radioactive ATP in the presence of a particular kinase for which we want to test the activity. And then we would observe that this protein target, this target protein or substrate, is converted into a phosphorylated protein by the presence and the activity of this protein kinase. So this is a very classical biochemical experiment. And this would immediately prove that this enzyme that we're studying here has the ability to transfer a phosphate ester group onto a protein, and we would then conclude that this particular protein has a biochemical activity that is that of a protein kinase. So this has been done, of course, for many proteins, but it is something that is really not generally tractable for all the factors of the proteome or components of the proteome, because we need to know what is the assay. We need to know what is the substrate. We need to know what is the anticipated reaction. Then we need to then carry out, is basically establish an assay, and we would of course then need to purify the protein that's to be tested. So to do that for a protein where there is nothing known, so a protein that might come out of a particular screen and we know nothing about this protein, to actually determine the biochemical function is a huge amount of work and generally not tractable on a proteome by its scale. So this is a problem because we are now, it's a fundamental problem because we are now able, we are extremely capable of measuring large numbers of proteins, and many of those proteins show interesting behavior in a certain experiment, but we don't know necessarily what is the function of these proteins, nor do we know how can we determine this function biochemically in analogy to this kind of kinase assay that I just showed. So many of the proteomic analysis where conditions are being combined or compared are usually represented in a plot which many of us, of course, probably everyone knows, which is this volcano plot. And we would test different conditions, for instance, tumor cells or non-tumor cells, or activated cells or non-activated cells, and we would measure the proteins in terms of which proteins change between these conditions, condition 1 and condition 2. And this plot then simply says we identified a large number of proteins. Each dot here is a protein. For the proteins that we have that are red colored, we are sure with a certain probability that we have correctly identified this protein, and that between the conditions, it shows a different quantitative behavior, so the abundance has changed between condition 1 and condition 2. This is a very frequent outcome of biological experiments where scientists try to figure out what is the difference between the proteome of two or more conditions. So we are able to do this, but then we would like to know, of course, what are these proteins doing? What are they telling us? What biochemical processes are different between condition 1 and condition 2? So we need to make the link between the identity of a protein to its function. The way we do this is usually by looking things up. So basically relating the proteins that are in this red part of the volcano plot and relate it to the known knowledge of the functionalized proteome. So it's actually remarkable how much is known about the proteome in the functional level, but many proteins, about 2,000, are still entirely uncharacterized. So this is about 10%. So we might say, well, this is not so bad because 10% is not a very large number. And one could say, well, eventually these 2,000, roughly 2,000 proteins, which are uncharacterized yet, eventually they will be characterized by and functionalized by some form of biochemical assay. So we make two assumptions that and I should say this is work that is compiled by the group of Amos Beroch, which maintains the SwissProt and UniProt databases, which are tremendous resources to look up what proteins might be doing in terms of their function. So we make two assumptions here, that the proteins for which something is known, that this information is correct and actually useful to use, and we would assume that eventually these other 2,000 proteins will also be associated with specific functions. So now I want to test these assumptions a little bit with some data from the literature. So let's first look to what extent the knowledge in the literature is spread out about around the proteome. So we can, this is work which is cited from this article here. We can analyze basically to what extent is the literature a even or representation of the knowledge spread out over the proteome, or to what extent is the literature biased on specific hotspots where the same proteins are always cited again and studied again. So basically, do we have the rich get richer situation where the same proteins get studied and published on this, a lot of papers get published on these proteins because already a lot is known, and or do we have a more even representation of the literature over a large number, ideally all the functions that the proteome can carry out? And so this is what the data show. This is basically an analysis of the literature with respect to a literature-centric analysis, where we ask what fraction of the literature is covered by specific proteins in terms of literature citations. So the red group here, to be explicit, the red-colored proteins are proteins that have more than 500 papers that report about these proteins. And we can see that these proteins that cover where more than 500 papers have been published on these proteins cover more than 75% of the total proteomic literature. If we add proteins which have 100 to 500 citations, or basically papers published on them, then these are probably close to 90% of the papers published on proteins are covered with those proteins where large number of reports have been published. These red and the green, however, when we go to a protein-centric representation of the literature, only cover about 25% of the proteome. So we can clearly see that there's a huge overrepresentation of relatively few proteins in the literature. And there is a large fraction of proteins, namely all these here, which the blue, the ochre, the gray, where there's less than 20 papers for about 40% of proteins, is less than 20 papers have been published about these proteins. So there's a huge bias in the literature today about the proteome. And so we could say, well, this is not too bad. We can simply expect that this will eventually even out. But it is currently a big problem, because when we look up proteins from these volcano plots that show up as with interesting behavior, then we typically are forced to go and look for proteins where a lot is known already, so the green and the red, and then we write something about these proteins in our paper and basically propagate, contribute to this bias. So this bias has been clearly noticed. This is an interesting report from the group of Stöger in 2018, where they said, can we take the protein literature and can we find features that are associated with these proteins, which would determine whether a protein is showing up in a publication. So this is basically the same question that one might ask in a biomarker study, where one has a number of proteins and would ask, do these proteins allow us to classify patients? So here we would not like to classify patients. We'd like to have features on these proteins that would classify basically the publications, whether these proteins are showing up in publications. And so what they came up with is that a relatively small number of features of genes or proteins predict the number of publications. So that means basically, and these are very well known, studied biochemical processes or cell biological processes, which have been extensively studied, and dozens or to hundreds of papers have been published. So this, I don't need to go through these names, but everyone who studies biological processes will know that these are parts of the cell biology which have been extensively studied, for instance, membrane proteins and specific biochemical functions. So they went even further. They even said, can we basically develop a classifier from the studying the proteins that are frequently appearing in the literature and make it through peer review, that would allow us to predict where, if you write a grant proposal, which type of proteins we should write the grant proposal apart, so that the grant proposal has a high likelihood of being funded. So they basically predict from the entirety of these proteins, where they try to extract features which predict that if we study these features, these proteins which have these features in a grant proposal, that we would have a better than random chance to actually get a grant funded. So this is a correlation with the predicted numbers and the actual dollars that would expended on that the model predicts that dollars would be expended on this based on these features, and that's the actual data that from NIH funding, and there's not a great but a fairly substantial correlation. So what this basically all means, that the publications that are making it through peer review and scientists publish are repeatedly focusing on similar features, and these similar features are covered by specific sets of proteins. So now we could say, well, this is probably temporary, because as the research goes on, we will eventually have all the proteins functionalized, and this would mean then that these biases will eventually disappear. So here, this is again from this paper from Sinha. They studied over time whether there were new proteins entering the literature. So new functions that are carried out by proteins that would enter into the literature. We have several curves here, and these curves are proteins that have a certain number of papers that report on them. So this one here is 500 or more papers. The green is 100 or more papers, and the curves up here, these kind of the two on top, are papers where there's virtually nothing known. So there's 0 or 1 paper that appears in the literature. So these are the newcomers. These are papers that report on proteins which previously have not been described. So this is encouraging, because from the 60s to 2000, and I come back to the reason why I made this bar here in 2000, is that in this biochemical age, there was every year a number, actually an increasing number of proteins entered the literature with description about their function that were previously not known. So there's an increase in the expansion of the coverage of the proteome in terms of function assigned to proteins. So it was kind of the hope that the bias would diminish. But now we entered in 2000, basically the omics age, and we see a trend, a distinctive trend, changing trend. The number of proteins that are reported newly in papers, so these are proteins for which nothing was known, and now someone went through the trouble of assigning a function to, for instance, biochemical characterization of this protein, and entering this in the literature has drastically decreased. So whereas the number of proteins for which 500 or 100 papers already have been published has increased. So the trend towards the bias, increasing the bias, the trend of the bias has been increasing as opposed to decreasing since the emergence of omics, the omics world. I think this is my personal view. This is subjective, not objective, is that the reason of that is techniques like clustering, volcano plot, association studies, basically data-driven approaches, where we generate a large volume of really high quality data in proteomics, but also of course in genomics and metabolomics. We then have the challenge to identify what these proteins or genes mean, and what we do is we go and look up what's already known and report on these proteins. It is much easier to write a paper on a protein like P53, where there is thousands of reports, and say we found a new aspect of P53, than to say to take a protein like this here, where nothing is known yet, and say we found this protein to react under certain conditions in its abundance, for instance, by when we stimulate a cell with a certain factor. Then reviewers will say, well, we have to go and learn much more about this protein. If we say it is P53 has a role in a new process, is as established by an association or by a volcano plot, then no reviewer will question that this is an important protein. So this is to me, this is fairly interesting, and I think it points towards an urgent need in the field of cell biology and biochemistry to reverse this trend and to find ways to enter new insights on a large, to report new functional insights on a larger number of proteins which have not really been functionally studied so well. Maybe there's very little known or nothing at all. This is also, of course, a great opportunity for proteomic researchers to undertake that, but it is difficult work, and it's not so clear how one would do that. So I want to summarize here this part. What I wanted to discuss is the determination of biochemical function of proteins that are identified in screens. For instance, in volcano plot screens, association studies, or in cluster, in analysis like clustering. So what I try to show is the experimental and functional analysis of proteins is not compatible on a proteomic scale. So we cannot just go and take 20 newly identified proteins that show interesting behavior in a association study like a GWAS study or PWAS study, or in a QTL analysis, or in a volcano plot, and go and say we want to study what these proteins are doing. This is too expensive, too slow, and too much work, and virtually no one is doing that. So then we focus the functional investigated on this of discovered proteins is mostly by literature reference, and we show, and I try to argue that the literature is blind to the function of thousands of proteins and is highly biased towards relatively few proteins from which there's already a lot been studied, and by reporting on these proteins new facts, we conclude we contribute to this bias. So and I even show through this interesting paper by the Stöger group that funding is focused, funding by, for instance, the NIH is focused on well-studied proteins. The rich get richer, and conversely, if you want funding from the NIH, you might as well study what is already known, which is a very depressing conclusion, of course, but it's actually supported by the facts, by the data from this analysis. Okay, so now
Rudy, just one question maybe from a participant. So in 1975, there was a spike where a large increase in publications of new protein coding genes, and then the spike went down. So can we comment on that? I think it's on the graph on the previous slides. Yeah.
Yes.
Thanks.
So this was around time here, around 80. I don't know for sure, but it could be that was roughly, I mean, you can see what happened there technologically, and this was, of course, roughly the time when gene cloning became feasible. And so probably, I mean, I don't know, they would have to look into this more, but it's possible that people started to clone genes with previously unknown function, express these genes so that one could then generate protein which could be tested. Because of course, when you want to undertake some kind of biochemical assay, one of the limitations is that the protein has to be available. And through these gene cloning techniques and gene expression techniques, where one could generate now amounts of proteins that are compatible with biochemical assays in large amounts, and also eventually with affinity tags, then this made, of course, many proteins much easier accessible to functional tests. So that would be my first guess, but I don't know whether this is actually the main cause. Okay, so now I would like to go a step further. So far I basically just illustrated the problem and illustrated also how difficult it is to assign a function to a newly discovered protein, or a protein for which nothing is known, to assign a function to it. The same is, of course, true for PTMs. And this is probably even a more pronounced problem with PTMs, because there the discrepancy of what can be measured and what's known already functionally is even larger. So currently, there's more than 100,000 human phosphosites alone have been described. And probably the estimates are that roughly 10% of them have some form of functional annotation. So there's a tremendous need to try to figure out what are these modifications doing, or how do they change a protein or the protein's function if they are present or if they're absent. So this is the topic I would like to discuss in the next few minutes. So we know that in proteomics or in protein chemistry, the same happens like in this transformer. There's two instances of pieces that are put together in some way. The pieces are exactly the same, but they're put together in different ways. So they can have different functional, also functional appearances. This one looks like a car would drive around. This is a kind of a monster robot that would have completely different functions than the part. So the same parts can be organized differently, leading to different function. And we also know that this conversion between one function and the other can oftentimes happen by changing the modifications. So basically, by driving the shape from one shape to another by a modification. So now translate this into the biochemical world. This is a graph. So this is work that was carried out by Tina Ludwig a while back when she was a postdoc in our group with Ana Paula Olivares and Paola Picotti. And this was a collaboration between our group and Uwe Sauer's group at ETH. The question was, can we identify modifications on enzymes that have a known function? And can we determine whether these modifications change the function of these enzymes? So this is the enzymes we studied. This is basically the yeast central metabolism, which is basically energy metabolism. We see here glycolysis, TCA cycle, and so very well known reactions. We initially, or this was Tina and Ana Paula and Paola Picotti, they initially identified the modifications they could detect by mass spectrometer on these proteins. So we see here that these are the reactions, these are the enzymes that have names that carry out a specific reaction in this metabolism. and they're color-coded. When they are phosphorylated, they are red. When they are acetylated, they are blue. And if they are thiolated, they are green. And we would, of course, like to know what are these modifications that occasionally, under certain conditions, occur on these proteins, doing to the activity of these proteins. So I wanted to ask specifically, does phosphorylation activate, inactivate, or not affect the catalytic activity of these enzymes in this metabolic system? So the experimental setup was that we grew cells, yeast cells, under various conditions, so 5 growth conditions, where the cells are put into different metabolic states, and 3 shift experiments where they're shifted from one state to the other. So the details are not really important, but when we think of this data matrix I showed before, this would be different conditions in which we measure the phosphorylation state of these proteins that are part of these metabolic systems. So we know the phosphosequence. We would then generate SRM assays. Today, one would do this with probably with DIA measurements, but the principle was to put the cells in different conditions with the assumptions that the metabolics, that these enzymes would be in a different functional state, and we would observe this functional state by doing quantitative analysis of the phosphorylation sites that are known, that were identified to be present on these proteins under the specific conditions. And then we would correlate this phosphodata with other data, and the data we correlated with was metabolic flux. And metabolic flux basically means is the flux of substrate through that particular enzyme. Each one of these enzymes has an adduct and converts an adduct into a product. And this flux is measurable also by mass spectrometry and by the field of metabolomics. So we basically would generate different data on each enzyme. We would determine the abundance of the enzyme. We would determine the phosphorylation state of the enzyme, and then we determine the flux, the metabolic flux through that enzyme under a variety of conditions. So this is a typical case where we generate this data matrix of conditions versus a quantitative entity, and in this case is the phosphorylation state, and we have correlated this then with the flux. So we can then do the following. It's an enzyme, hexokinase, doesn't really matter, the name doesn't really matter, but it's an enzyme that carries out a specific function. It carries out glucose, it converts glucose into glucose 6-phosphate with the utilization of ADP. So when this enzyme, we can measure the phosphorylated enzyme form of the enzyme by proteomics. We measure the flux through this enzyme metabolomics, so basically how often it does this as a function of its phosphorylation state. And if this correlation is positive, then that means the more phosphoform is present, the higher the flux. We would say the phosphorylation activity, the phosphorylation event activates the enzyme. When the flux correlates positively with the non-phosphorylated form, we would conclude that the phosphorylation actually inhibits the phosphorylation, because the more non-phosphorylated proteins present, the higher the flux. So we can use this correlation of 2 data types. We integrate this data by correlation, and we basically say we can conclude whether a phosphorylation event that's observed in any one of these enzymes is activating, inactivating the enzyme, or is doing nothing to the activity of the enzyme. So this is what came out. These are names of enzymes in this pathway, in this central carbon metabolism, that are for which we found phosphorylation sites. And here we have a number of them where phosphorylation activates the enzyme function. We have a number of them where the enzyme is inactivated by the enzyme function. And so these are relatively few enzymes, and there's in the whole study were a few more. So in fact, there were 16, we found 16 phosphopeptides that were could be tested in this approach. So you might say, well, 16 is not really a proteome-wide study, and so this is of course true. But before this was done, this was not so long ago. This was like between 12 and 15. There's only one, the whole literature of metabolism, which is one of the best studied biochemical systems that we know. There's only one enzyme for which the phosphorylation has been described to affect the enzyme activity. This was the enzyme PDA1, and so in this simple study, we could create data on we could discover 7 phosphosites that activate enzymes, 2 that inactivate enzymes, and several others had no effect on the enzyme activity. So this is not large numbers, but it expanded the number of the prior knowledge multiple times. And this, of course, was done. This was done by SRM by following a relatively few phosphorylation events. We have now since repeated this analysis with a much larger number of phosphosites monitored over a larger number of conditions with DIA measurements. And these data have been acquired, but they're not really analyzed yet fully. But it doesn't matter, because I just wanted to show a principle that we can use data-driven inference by relating the data obtained in systematically structured assays to relate to some other activity, for instance, catalytic activity that can be measured by, for instance, metabolic flux analysis to learn the functional significance of specific PTMs. I think this general approach is extendable to many other biochemical systems, and I think this circumvents the need to establish a definitive biochemical assay for every protein in tedious one-on-one work. So the summary here would be the inference of functional states of identified proteoforms. The abundance of a protein does not indicate the amount of its activity, as we know that, of course, and we would like to know whether the activity is modulated by specific proteoforms, for instance, phosphoforms of a protein. Direct activity measurement is prohibitively expensive on a proteomic scale. It should be a scale. And activity state can be inferred by correlating suitably structured data sets. The example I chose here or presented is protein phosphorylation, quantitative changes over a number of conditions correlated with metabolic flux changes under these conditions through specific enzymes. And so these are basically association studies, and they depend on high quality protein versus abundance data matrices and some other type of data, for instance, metabolic flux analysis. So we're doing time.
20 minutes,
how are we doing
roughly 20 minutes left.
Okay, so now I would like to move to this arrow here and focus on the function of protein interactions and specific protein complexes. This is, of course, an interesting field, because we know also from biochemistry that most biochemical functions are not carried out by a specific protein in isolation, but most frequently by complexes where multiple proteins associate with other proteins, or for that matter, RNA. And we also know that these complexes are frequently modulated, for instance, by PTMs, so for instance, by phosphorylation. And so there's this argument which was used a lot by the argument of guilt by association. So if you know some proteins, a protein of a complex to have a specific function, for instance, to be a protein kinase, and you can establish that the green protein associates with this protein kinase, you would assume that this green protein in the wider sense is involved in protein phosphorylation as a broader biochemical activity. So this is the topic I'd like to now discuss. I would like now to discuss exemplify with a study that a huge amount of very useful information can be learned from the protein complex level, so how proteins are organizing the protein into modules. I'd like to show that these modules are to some extent a buffer of changes that are occurring here at the transcriptional and translational level in terms of abundance changes. And I would like to show that we can detect changes in the proteome at the level of their associations, the modularity of the proteome, that are directly related to phenotypic changes that we can observe. So let me first explain what we do here, and then I show some results. To test the idea that at the level of the proteome organization into modules, there is important, and changes thereof, important biochemical information, functional information can be learned, we need to first find a way to perturb cells so that they might change the molecular landscape. This, of course, can be done by directed change through CRISPR, for instance, or any other genetic manipulation. We like to use naturally occurring variability because we know that this variability is not lethal because the cells have survived, and so that usually this naturally occurring variability is relatively mild, and because it is not lethal, but it is also certainly measurable, and we can measure the propagation of for instance, genetic variability. So we chose here to use cells which are inherently from the same source. This is HeLa cells, which are very frequently used. And we chose these because we assumed that over time they had undergone genomic drift, so that they have changed the genome, and then we can measure how these genomic variants percolate along the axis of gene expression, along the axis of the central dogma. We generate multi-layer omics data sets from a panel of HeLa cells. And then we compute how the genomic variability affects the transcriptome, the proteome, composition, and the organization of proteins, and the function, as expressed by some phenotypes. So this is a graphical abstract. We selected from different laboratories HeLa cells, which have been cultured in their laboratory for a certain time, assuming that they had undergone genomic shift. So these were 14 cell lines that we collected. Some of these cell lines were collected only a few months apart, like 2-3 months apart. From each one of these cell lines, which were grown under the same conditions, we created genomic data by measuring the copy number variation, namely how the genome had altered, not by single nucleotide changes, but by whole blocks of the genome that have been either duplicated or deleted. We measured RNA expression, steady-state proteome, protein turnover, and we measured some phenotypes. So these data all exist, and it were collected from all of these cell lines. And here is also to show that the cells are certainly different, even though they have the same name. This is the doubling time from these 14 cell lines, and this was hours. So we can see that the fastest growing, fastest dividing, under the same conditions, same medium, same person, same laboratory, would grow, divide roughly twice as fast as the slowest one among these collected cells. So there's clearly a phenotypic difference. And then we measured another phenotype, and that's the ability of the cells to be invaded by Salmonella. So Salmonella are bacteria that infect human cells, and so one of the phenotypic measurements we could easily undertake is to ask which one of these cells, if we fed them or exposed them to a certain number of Salmonella, how many of these Salmonella would go into the cell. And so basically the ability of the cell to be infected by the Salmonella is a quantitative phenotype that could be measured. And this just shows that these cells also differ substantially in this phenotype. Basically, this is the size of this graph here is related to the ability of the cells to be infected by Salmonella. So they differ quite substantially. This one cannot be infected at all, and the others can be infected to some extent, but to different extent. So these are two phenotypes. This is proteomic data, and this is genomic data now to illustrate the difference, the genomic difference at the level of copy number variation. So we see here, each circle, concentric circle, is one genome, is the genome of one of these cells. Red means there's a high copy number. So there's multiple copies of this particular locus present. Green means a low copy number, like 1 or 2. And so we can clearly see that each one of the cells has a different copy number profile. So these are all relatively similar here, but then these are different in this chromosome. And so everyone will see graphically that they're different. I'd like to point to these two outermost cells, 12 and 14. This area here and this area here are substantially different. And these cells were grown in the same laboratory and are harvested about 3 months apart. So within 3 months of culturing these cells in the laboratory, a substantial duplication or loss of duplication of this chromosome 15 and chromosome 8, a substantial part of it, was observed. So these are highly unstable cell lines which have implications for reproducibility of research results, which I don't want to discuss here. We simply use this here as a system where natural variability has occurred through genomic instability mechanisms, and we want to ask, how does this genomic variability affect the proteome and the organization of the proteome? So this is an overview of relative gene expression patterns in cell lines for 2 chromosomes at the level of copy number, genomic, transcriptomic, and proteomic. We can immediately see that very strong blocks here of increased presence, copy number presence of specific chromosomes or parts thereof is tendentially also apparent at the transcript and protein level, but not as distinctively. So even in this red zone here, we have green zones, which means there is proteins and transcripts and proteins in loci which are amplified that are not amplified. So there's not a strong correlation between the copy number, the gene dosage, the abundance of the transcript, and even less so for the abundance of proteins. So this is graphically shown here. This is shown here by actual numerically by measuring the correlation between, for instance, copy number and mRNA, copy number variation, and protein abundance. We see always a broad distribution. We see a mean correlation factor which is positive, but not strongly positive. So basically, this means that, which probably everyone already knows, that there is not a predictable correlation that's strong enough to allow us to predict the amount of protein from the amount of an RNA, and does not allow us to predict the amount of RNA from the amount of copy numbers in a gene. So this is pretty well known. And now we wanted to see whether we could deduce some conclusions in relation to the proteome organization. So now
a clarifying question on the previous slide. What is a protein loss at the bottom?
Oh, this is the degradation. K-loss, this is basically a representation of the half-life of the protein, which is measurable by mass spectrometry, by using isotopic labeling, and then basically measure how the signal decays. So it's a representation of the half-life. I'm coming to this. I'm glad you asked that, because I use this term now in the next slide.
Thank you.
So now we correlate So now this is going a step further. Now we want to ask the question whether in the proteotype or proteome organization, namely the fact that proteins associate into complexes, has any is a determinant, or can be whether the gene dosage that we see can be compensated by the organization of the proteome into protein complexes. So to explain that, or to investigate that, we categorize the proteins that were measured into two groups. One is a group which we call complex in. These are proteins which are known to be in complexes, and they know that because there are proteins which are known to be in a complex in a database called Quorum. This is the best curated database for protein complexes. And we have, so these are the complex in proteins. And this one, the greenish ones, are the complex out proteins. These are proteins which are not known to be in a complex. They could be, but there's no evidence for it at this point. So we then can correlate the copy number variation for the complex in and the complex out proteins between copy number and mRNA. And we see that there is no difference between them. There's this distribution for copy number to mRNA correlation, which I showed in the previous slide, but there's no difference in really that between the complex in and the complex out transcripts. So that means the transcripts do not know, of course, whether the protein eventually will end up in a protein complex, and so there's no signal that would indicate that the eventual organization of the projects in those proteins into modules is not apparent at the level of the transcript. This is different when we correlate copy number with proteins. Here we see that there is a clear difference between the median here, correlation, but basically the distribution of correlation to median for the complex in and complex out proteins. And the opposite is true when we, well, they're also different, but in the opposite way, is when we correlate the copy number with the K-loss or degradation rate of proteins. So basically the way these results can be explained, that if we go along the axis of gene expression, the cell expresses protein transcripts, and these transcripts can be in these groups that eventually we call complex in and complex out groups, but at the level of the transcripts, this is indifferent for the correlation between copy number and RNA between the two groups. When we go to the proteins, there's a clear difference, and the proteins that are in are down. The correlation is lower generally than for the complex out one, so they're down buffered in terms of their strength of correlation. So what we conclude here is there's proteins synthesized which cannot be incorporated into a complex because the other members of the complex are not present. These proteins have no place to go, and they're preferably destroyed. And we see this because they are from this picture here that they are preferably degraded. So we will conclude here that the organization of the proteome into functional modules is a mechanism by which amplitude changes coming from genetics or other perturbations is buffered, and that this is a stabilizing factor from the point of view of functional perturbation, of moving molecular perturbations, functional perturbations. So this enticed us to try to start looking whether we could measure changes in the organization of the proteome systematically. And we do this by a very old technique, so we basically would like to have a cellular network. We would have a state 1 and state 2. This could be one cell line from this HeLa, for instance, another cell line, or an inactive and an active cell line, or whatever the case may be. And we would like to now project these abundance changes that we see onto this interaction net, protein-protein interaction network. And we'd like to say, not only are these proteins changed in abundance, but they're also changed in their association into specific function units. And because oftentimes the function of a molecule in a complex is known, we would like to maintain that associating proteins in complexes would help us to functionalize proteins, even though we cannot do a direct biochemical assay. So the experiment we're doing is a very old experiment, but it is now very tractable and actually highly informative through new techniques. So we isolate native complexes, we extract them under mild conditions, which is basically was the mainstay of all biochemistry in the 60s and much of the 70s and even earlier. We separate these complexes, native complexes, by size exclusion chromatography, which is also the mainstay of protein purification, except this is now done with very high quality, high pressure columns, and we collect about 65 fractions, consecutive fractions. And we analyze each fraction by DIA mass spectrometry. This generates elution profiles across this native separation for approximately, from a cell line for approximately 5,000 proteins. And we know, of course, the molecular weight of the monomeric form of this protein, so we can immediately predict whether a protein is monomeric or it's associated with something else. And through a software tools that were developed by Isabel Bludau and Moritz Heusel, we can do hypothesis testing whether a certain protein complex is present or not. So this was described and is not the subject of this discussion. Now I'd like to go a step further and basically start with about 5,000 such elution profiles, and we project them now onto protein interaction networks. We basically test the hypothesis whether a specific perturbation changes a specific region of the protein-protein interaction network. And the protein-protein interaction networks we use is quorum, string, or PrEP. These are all different types of protein-protein interaction networks that have been generated. It also could be bioplex. So we represent here a protein interaction networks by having proteins, these symbols, and they have an edge, and this edge indicates the likelihood that in the data set, the seq-SWOS data set of a specific cell, this interaction is observed. And this interaction, we call this interaction observed if the proteins, these two proteins, that this half moon and the blue precisely co-elute in the dimension of native separation. So there's approximately a dozen factors that tell us whether they are likely to be interacting, because it's not just the coelution of the apex, also the peak shape, the relative intensity, and so on. So it's basically the same principle that we apply, that you would apply with in Skyline for scoring peak groups of transitions to identify or determine that the peptide was present. There's a number of factors which allow us to calculate the probability that this interaction is observed in this state. So we now have two states, for instance, a state A and a state B, and we would say this interaction here is observed in this state, it is also observed in this state. We have also interaction here, which is between the ochre half moon and the violet circle, and in this case, there's evidence that in this state that this interaction is present, is observed, and in this case, it's not observed. So basically, these peaks are absent, they don't co-elute. So with that, we can walk through basically the whole interaction network, a string, and test each interaction, whether there's evidence in the data set that in this particular instance, this interaction is likely to be active or not. So this is an algorithm that is called CCAT, was developed by George Rosenberger, who is now a postdoc in the Califano group, and it's been described in bioRxives. So now when we use two of these cell lines, of these HeLa cell lines, that are phenotypically different, we can see differences at the level of protein organization, which are fairly profound. And I will show then that they directly lead to functional insights that relate to phenotypes. This is an overview Each dot is a protein complex, they're color coded, and in this complex we see various differences. The color code is down here. We see differences in total abundance, in monomeric abundance of this protein, in the assembled abundance of this protein with other proteins, and so there's a whole range of different qualitatively different differences we see for proteins that are part of protein complexes. Overall, we can test in this data set about 7,000 protein-protein interactions using this algorithm. And this is a balance sheet of the type of differences we detect by two cell lines which are nominally HeLa cells, but they have diverged through genetic drift. So we see, for instance, 86 complex stoichiometry differences, 82 differences in protein interactions, 80 differences where the complexes of different abundance, and so on. So there's a rich information that is directly functional relevant because each one of these complexes that are undergoes a change very likely has some biochemical connotation, for instance, from APMS experiments or from association studies. I think I need to skip that for reasons of time.
Yeah, we have just 1 or 2 minutes left.
Yes, but I wanted to show now that specific changes in complexes can explain the phenotype of invasiveness of Salmonella, because we can basically reconstruct from this data a whole organelle, which is called the invadopodium, which is a complex mechanism of where the Salmonella are invading the cell, and if there's a disbalance of subunits, which is in a complex which is observable from this data, this organelle is not forming, and the Salmonella cannot invade. So basically, to make a link through the genomic variation to the expression of genomic variation at the level of transcripts and proteins, but now also at the level of protein interaction networks. And because proteins tend to interact around specific functions, we can then relate some of these perturbed functions to specific biochemical pathways. For instance, this invadopodium, which explains a numerically measurable phenotype. So I was planning to, but now run out of time, to also make the point that we can detect differences in individual proteins, for instance, through alternative splicing or PTMs, and then test whether these modifications would affect the organization of the proteome. So specifically what we're trying to test here is whether if a gene generates alternatively spliced protein through alternative splicing different proteins, whether these different proteins associate with different complexes. That would indicate immediately that the two or more splice proteoforms that are being generated undergo or participate in different functions. And this is the principle of that is that if we have a protein showing here the exons that can be alternatively spliced into a blue protein where this exon is being used, and this one where this exon is being used, we can use in the native separations of these protein profiles through Sec fractionation. We can ask whether the reddish and the blue exons basically form proteins that associate with the same or with different protein complexes. And so Isabel Bludau has generated an algorithm that tests that for the whole data set. And so she can detect from, in one particular instance, where we look for cell cycle dependent changes in proteoforms associating with different complexes from about 4,800 proteins. She detects 413 instances where a gene generates different proteoforms, and these proteoforms associate with different protein complexes indicating different functionality of these spliced isoforms that are being generated, some of which are cell cycle specific. So I don't have time to show this data, but I would now like to summarize what I try to show and try to discuss. So initially I showed that proteomics has made enormous progress from the point of view of identifying proteins from virtually all protein coding genes. I then tried to say that we're facing two very significant challenges still in proteomics. Even though we can measure these proteins very well, we would need to understand and eventually factor in that the protein coding capacity of the genome is vastly expanded across the axis of gene expression through a variety of mechanisms, including splicing, including PTMs, including to some extent combinatorial association of proteins into modules. And that it is this complexity that determines eventually the phenotype. I then also try to show the other fundamental challenge we're facing is that even though we can measure a lot of things along this axis, we don't necessarily know what these molecules or modules are doing biochemically. And I try to focus on a variety, on several levels. For instance, that we would be able to use correlative analysis to functionalize PTMs. I try to show that there's enormous biochemical functional information at the level of the protein organization. And at the last part I wanted to show, which I had to basically skip over, that we can combine the association of proteoforms, differential association of proteoforms with different complexes as a readout to say whether specific splice forms or PTMs participate in different functions by associating with different functional complexes. So with that, I would like to finish and acknowledge my collaborators and group members whose work I discussed here. Everything that we do and presented is based on data sets which are mostly generated by this SWOS-DIA method, which was developed by Ludovic Schier and Pedro Navarro. And Hannes Röst and George generated software tools. I mentioned the collaboration with Matthias Mann's group and Brucker on this DIA-PASEF instrument. Hannes Röst is also involved in that by extending the OpenSWOS software to the extra dimension of ion mobility that is provided by this DIA-PASEF. I mentioned discussed the functionalization of phosphoproteoforms, work of Tina Ludwig, Ana Paula Oliveira, and Paola Picotti. I mentioned complex protein-protein interaction network differential analysis, the work from Isabel Bludau, Moritz Heusel, George Rosenberg, and Yansheng Lu. And the association of proteoforms with complexes, the work of Isabel Bludau, Max Frank, Charlotte Ma, and Hannes Röst from Toronto. You see that virtually everyone I list here has now a different name, a different organizational affiliation, and that is because our lab basically dissolved, and all these fantastic scientists who made these contributions are now continuing their career in different institutions, which makes me, of course, very happy. Thanks for listening, and I'm happy to answer some questions.
Thank you so very much, Rudy. So on behalf of everybody, have a big virtual round of applause for you. So we do have a bunch of questions which I left to the end because some of them are fairly general. So we have a series of questions, and we will take a few so we won't go for too long. So a few technical questions about the network. Do you get the same networks if you use S200 and S300 chromatographic seq columns?
As far as the proteins complexes that are separated by these different columns, the answer should be the same, but they have different ranges of separation power, and then of course there is a number of complexes that you don't see in one column but may see in the other and may be resolved. So the S usually refers to Cephadex, And we don't use Cephedex. This Cephedex, of course, is extremely well established, but these are tend to be relatively large columns, and these are low pressure columns. We use high pressure columns to do basic HPLC type separation by sizing, and of this there's a whole panel of columns available. Some separate up to 10 megadaltons, or even very large modules like mitochondria are still separable. And in fact, we actually do see not mitochondrial ribosomes, but in fact we do see flavors of very large particles like ribosomes, which is a very interesting direction of research, which is very heavily pursued. That modules are extremely well studied biochemically, like the proteasome, the ribosome, and others, COP signalosome. They are not inert modules. They actually appear in different flavors, and there's a lot of biology to be discovered there. And I think these native separations are a good tool to do that.
So another question is, how do you assign proteins to complexes? Are there any quantitative value cutoffs that you use?
Yes. So we assign. So it's of course probably well known that the idea of separating these proteins by native complexes and then mass specking through each fraction is of course not new. There's a lot of work that's been done by many groups, particularly Leonard Foster, Angus Lamont, and others. Usually the problem was not the measurement, but exactly the problem that the question raises. You have roughly 5,000 protein elution profiles along the size exclusion axis. The size exclusion chromatography is not a highly resolving technique. It has a peak capacity of certainly below 100. So peak capacity is basically an indication of the number of bins that you could create to really distinguish different molecular entities from each other. So it is essentially a problem that you have, by chance, a relatively high likelihood that out of these 5,000 proteins, 2 might co-elute in the same peak, but not actually interact. So this has been a difficult issue for a long time. And so we proposed that we basically would make a transition like we made from discovery proteomics to targeting proteomics, which is basically a transition from discovering things to doing multiple hypothesis testing. And we would do this also at the level of protein complexes. So we call this complex-centric analysis, and we use prior information from for instance, from a database for a quorum or string, and then we test the hypothesis whether a interaction that is known to occur is present in the data set that we have generated. So this narrows down the space a lot and basically converts the problem into a multiple hypothesis testing problem for which there's formalisms to also calculate probability values that this observation is correct. Whereas if you're in the discovery mode, it is much, much harder to do. So we think that this transition to a hypothesis testing mechanism has been very beneficial for the analysis of protein complexes and protein interaction networks, as I tried to show, because we can assign to each interaction a probability which we believe and have evidence through testing that they're reasonably accurate. And so the false positives are basically are controlled or are going away. The downside of this approach is, of course, that you do not discover new interactions which have never been observed. And so we think that the benefit of having probability values associated outweighs the danger or the threat that you lose or miss some interaction which have not previously been discovered.
Okay, one more question. There have been several reports indicating the importance of peptides or small proteins, such as those codified by small ORFs. They are often not detected in traditional proteomic analysis because they're not in the databases, they're not detected, etc. Using the current technology, how can we deepen in this so-called hidden proteome?
Yes, of course, there's many groups have worked very diligently to try to identify these smaller molecules, smaller proteins. Technically, from the measurement point of view, this is, I think, quite reasonably well under control. I mean, there's ways to select smaller fractions, to do size separation, for instance, to fractionate small fractions, and then to sequence these molecules. Also, of course, that's an area where top-down mass spectrometry is particularly powerful because we're dealing with relatively small molecules. And so I think from the measurement point of view, again, this is a reasonably tractable problem. The real issue, again, is what I try to discuss through my whole presentation, is to try to make biological sense out of this. So, for instance, there has been a lot of effort for quite a while to measure blood plasma, small molecules. I mean, small peptides. And from the point of view that they might develop into biomarkers. It turns out this is very difficult to do. Not, again, of course, also from the measurement side, but even more so from the interpretation side, because there's a lot of factors that determine whether such small molecules, small proteins are present and why they are present. Many of them are thought to arise simply from unspecific proteolysis, basically be proteolysis products. Others may be designed to be cut out specifically through processing to create a new functional entity. There's cases of that known where a polyprotein is cleaved into smaller polypeptides. Each one of them has a specific function. But to learn whether they are functional, whether they are simply byproducts of degradation or byproducts of some other process, is actually very difficult to do. And I think this is, again, an example where the measurement has progressed very quickly. The assignment of biological significance has lagged behind because it's a particularly challenging problem.
Well, thank you, Rudy, very much. So I think in the interest of time, we need to stop. We have quite a few more questions. So I'm carrying them over to the Google Doc. And Rudy, if you have the chance, if your schedule allows, maybe take a look at those if you can answer. But if not, we certainly understand. So let us close.
I make an effort to go through and answer as many as I can in the next.
Perfect. This is perfect. Thank you so very much. So if I didn't get the chance to ask your question, it will be posted in the Google Doc. Well, thank you very much again to Rudy, to our speakers, and to all the participants for being with us today. I hope that it was a good use of your time. And so tomorrow and Friday, we have a deep dive into Skyline with presentations led by Brendan McLean. We are very fortunate to have a chance to follow these presentations by the developer of Skyline. And it will be fairly technical, fairly in depth, and I think we'll be very fortunate to have a chance to learn from Brendan as well. Thank you so very much for joining us today.